The dance was named Balumpa dance, and it told the story of a group of girls while singing a folk and singing, and also being accompanied by music. They joyfully sang and danced together. And for the last dances, this is from the Tolaki ethnics. We call it Lariangi dance. And also they will perform Salulo dance. Lariangi dance is a dance of Tolaki people that is performed for guests of honors and as a form for respect for visiting Tolaki Island. And now is showing Lulo dance. It is a sign of togetherness and unity of the people in Southeast Sulawesi while holding their hands to unite regardless of nation origin. Okay, please big round of applause. It was so attractive performance. Thank you very much for DNR Kandari dance. The honorable distinguished speaker, ladies and gentlemen. Continuing, we are going to have chanting prayers led by Mr. Apotheker Mulyadi Prasetyo as from. So Mr. Mulyadi, the podium is already in front of us, so we invite you.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, let us bow our heads for a moment, pray to the God Allah subhanahu wa ta'ala, so that our international seminar can be precious to all of us. I will lead this prayer based on the teachings of Islam, and those who are not Muslim, you are pleased to pray according to each of your beliefs. Bismillahirrahmanirrahim. Alhamdulillahi Rabbil Alamin. Wassalatu wassalamu ala Rasulillah wa ala alihi wa sahbihi wa man wala. Allahumma inni as'aluka ilman nafi'an wa rizqan tayyiban wa amalan mutaqabbalan. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatan min lisani yafqahu qawli. Rabbana zalamna anfusana wa illam taghfir lana wa tarhamna لنكونن من الخاسرين. ربنا اغفر لنا ولإخواننا الذين سبقونا بالإيمان ولا تجعل في قلوبنا غلا للذين آمنوا ربنا إنك رؤوف رحيم. ربنا لا تزغ قلوبنا بعد إذ هديتنا وهب لنا من لدنك رحمة إنك أنت الوهاب. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار. وصلى الله على نبينا محمد والحمد لله رب العالمين. والسلام عليكم ورحمة الله وبركاته.
Waalaikumsalam warahmatullahi wabarakatuh. Sadaqallahul azim. Ladies and gentlemen, now we are going to listen the opening speech delivered by the head of Pharmacy Department, Ibu Apoteker Waude Yuliastri SPharm MSc. Please welcome Ibu Apoteker Waude Yuliastri SPharm MSc.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Let us say Alhamdulillah at this wonderful event. Praise to be God for providing graces, kindness, and help for us to meet on this special day. Firstly, I would like to thank and welcome to honorable ones, the founders of the Universitas Mandala Waluya, Director of Universitas Mandala Waluya, professors and lecturers from various universities who have taken the time to attend our conference today. Secondly, I would like to say thank you to the founders of the Mandala Waluya Foundation, the Dean of the Faculties of Pharmacy, Waluya University, the Head of Pharmacy Department, Bina Husada Polytechnic, the Head of the Pharmacy Department, Stikas Pelita Ibu, the Head of Kendari Regional Board of Indonesian Pharmacian Association, the Head of Kendari Branch of the Indonesian Pharmacists Association, the Head of BPOM Kendari for attending our international seminar invitation. I would also want to thank and welcome our presenters who have accepted our offer to present at this virtual conference. We also extend a warm welcome to the participants of our second international conference. I want to thank everyone who has joined us, both in this place and virtual. In the second on Mandala Waluya International Conference on Pharmaceutical Science and Practice 2023 with the theme Current Technology in Pharmaceutical Science and Research. We as healthcare professionals and organizations involved in the pharmaceutical sector believe that it's important to discuss the latest trends and challenge in pharmacy practice and pharmaceutical science. Through this speech, I, as a representative of the Department of Pharmacy and the Faculty of Science and Technology at Universitas Mandala Waluya, apologize for any shortcomings during this activity and hope this meeting will be beneficial for all of us. Thank you very much. Best regards.
Great. Thank you very much for the head of Pharmacy Department of the Mandala Waluya University. Excellencies, distinguished guests, and participants. Now we are going to listen to the opening speech delivered by Director of Mandala Waluya University. Also, we would like to ask respectfully the willingness of Rector of Mandala Waluya University to officially open this international conference. To all the participants, please welcome in our screens, Ibu Dr. Ratna Umi Nurlila, SSC, MSc.
Thank you so much. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to convey my greeting and appreciation to all of the native speakers, especially Professor Taifo Mahmud, Orange State University, Professor Apoteker Mutaharidi, PSD, University Pajajaran, Dr. Suman Cinta from Institute of Pharmaceutical Research, University, Asep, Professor Dr. Nijib, from University of Malaysia, Amira Muhammad Ghazali, PSD, RPH, from University Sains Malaysia. Thank you for you participant in our event greeting for Mandala Waluya University. Good morning, ladies and gentlemen. On behalf of Rector of Mandala Waluya University, I would like to declarasi that second Mandala Waluya International Conference on Pharmaceutical Science and Practice is now open. Progress in research and invention in the world of pharmacy practice and pharmaceutical science is dynamic and rapidly improving. A scientific meeting to bring together worldwide experts to discuss and later trends and colleagues in pharmacy practice and pharmaceutical science in consequently necessary. To facilitate the expert meeting and sharing knowledge among the pharmacy department of pharmacy, Faculty of Science and Technology, University Mandala Waluya will hold the second Mandala Waluya International Conference on Pharmaceutical Science and Practice, MW-ICPSP 223, with the theme Current Technology in Pharmaceutical Science and Research. I hope this webinar will accomplish its aim and early decide that participants will be able to benefit from the presenters and discussion, and the webinar will edit and development of pharmaceutical science, not only in Indonesia, but also in worldwide. I would like to thank the organizing committee for the tremendous effort to make the program come to the realization. I hope all of the speaker and participant will again many benefit, hopefully. We will meet again in the next MW-ICPSP program. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh. Thank you. Thank you very much.
Okay, the keynote speaker is will leading by Dr. Apoteker Rifatul Mahmuda, SPAM MSc. Before we continue, for our first keynote speaker, Professor Taifo Mahmud, I'll read about the Dr. Apoteker Rifatul Mahmuda, SPAM MSc CP. She is a lecturer at Mandala Waluya University. in the Department of Pharmacy. Ibu Dr. Apoteker Rifatul Mahmuda SPAM MSc finished her bachelor degree at UIN Alauddin Makassar, then got his apothecary, General Ahmad Yani University, then continue master degree and PhD degree at Institut Teknologi Bandung in the field pharmacology in a pharmacy clinic. For Ibu Dr. Apoteker Rifatul Mahmuda, we invite you in our podium. Okay, ladies and gentlemen, for this first session, the keynote speaker is Professor Taiful Mahmud, and this session will be in the three sessions. The first one is presenting by Professor Taiful Mahmud, and then it's a Q and answer. Then we would like to ask for Dr. Apothecary Fatul Mahmuda to give the symbolic certificate after the keynote Q and answer session. Okay, thank you.
Oke, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Respectable to Chairman of Mandala Waluya University, Honorable all to the lecturer of Department of Pharmacy, Faculty of Science and Technology, Universitas Mandala Waluya Kendari, and Honorable all the participants of this conference. Ladies and gentlemen, I am Rifatul Mahmuda, moderator in this session. I am very pleased to see you all here and welcome to this conference. The theme of this day is current technology and pharmaceutical science research. Before the presentation begin, I will inform how the presentation will be going on. First, each speaker presentation the material about 50 minutes, and then second, question and answer section about 50 minutes. So if you want to ask the speaker, in each speaker, you can write the question in the column chat via Zoom Meet. And we selected three questions. Okay, before we start to let the speaker to the presentation, let me introduce the presenter or speaker in this session. First is Professor Taiful Mahmud. Let me read the CV. Professor Taiful Mahmud was graduated from University of North Sumatra with Bachelor Science degree in 1989 and apothecary in 1991. He's continued his MSc degree in 1994 and PhD in 1997 at Osaka University. He's continuing his as postdoctoral research associate in 1997 and 2000 to 2000 with Professor H.G. Floss. In 2000 to 2003, he's becoming an assistant professor in University of Washington. His career started as an associate professor in the Department of Pharmaceutical Science in 2003 to 2007 at Oregon State University. He obtained it as an associate professor in 2007 to 2030 in Oregon State University. He currently is an honorary professor of natural product and medicinal chemistry. He has published 122 papers, is EC Index Journal. Then Professor Taifo has more expensive experience research that focusing in natural product. Besides, during discovery and development, his group employs a multidisciplinary approach that utilizes cutting-edge technology to product novel pharmaceutical. Some recent upliftment of this group research including discovering new families of enzyme and development anti-malaria, anti-cancer, sunscreen through genetic engineering or synthetic biology. Okay, the second our speaker is Professor Apoteker Muhtaridi, PhD. Professor Muhtaridi has graduated from University Pajajaran, bachelor degree in 1998, obtained his MSc degree in 2004, and at IPB University in food analyzing, and his PhD degree in enduring design and simulation in 2013 at University Science Malaysia. He started his career as ASEAN CSDD as advisory board member in 2018 until now. He has the founder and honorary member of Indonesia of Bioinformatics and Biodiversity Association Science 216 until now. Then he also the head of scientific and publication affairs of Indonesian Medicinal Chemistry Association Science 218 until now. He has the sub-member of Collegium Pharmacy Industrial. He has head of advisory board of West Java in Indonesia of Pharmacy Association Science 218 until now. Then he currently is a honorary professor and head of Department of Pharmacy Analysis and Medicinal Chemistry at University of Pajajaran. He has published in last 10 years for 55 papers, and he has 12 national accreditation journal. His H index is 10, and his H index polar is 30. He has 6 patents and have an 8 book chapter. He has been invited a speaker and keynote speaker in scientific meeting in the last 5 years. He is reviewing in manuscript journal Science as Educational Ministry Journal, the editor inside of IGPST, the editor journal in Mandala Pharmaceutical magazine, as editorial member of Malaysia Journal of Pharmaceutical Science in University Science Malaysia, and member of Erudite Journal. He has he has experience research in drug discovery development from natural product, pharmaceutical analysis for natural product compound. Okay, before we will be going on to the presentation, I will invite the first speakers, Professor Taifo Mahmud. Are you here?
Yes, thank you for the introduction.
Okay, how are you, Professor Taifo?
I'm doing good, thank you.
Okay, let me invite the Professor Taifo Mahmud to present the material of the presentation. Professor Taifo Mahmud, time is yours.
Right. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. So let me share my slide first. All right. First of all, I would like to thank the organizer for inviting me to talk in this conference. So my name is Taifu Mahmud. I'm from Department of Pharmaceutical Sciences, College of Pharmacy, Oregon State University. So Oregon State University is a public university in the state of Oregon on the west coast of the United States. So this is, before I start, I just want to show you the Oregon State University Corvallis campus. So this is the middle of the campus. Actually, it's quite big campus and pretty beautiful. And the College of Pharmacy actually is not in the picture on the left side of this campus. This picture, and then my lab is somewhere around this area. So today I would like to talk about biotechnology approaches in natural product drug discovery. So this is the outlines. We'll be talking about the introduction, and then biosynthetic studies and production of new natural products via using genetic engineering. And then I will be talking about genome mining and the discovery of new natural products, and then how we increase natural product production using molecular genetic approaches, and then how we produce new natural product analogs by chemoenzymatic method. So as in the introduction, as many of us know, that natural products continue to be an important source of new drug leads. If we look at all small molecule approved by the FDA as drugs from 1981 to 2019, about 1/3 of the drugs that have been approved, they are either natural products, botanical or natural product derivatives. Another 1/3 actually a synthetic compound, but still they are inspired by the structure of natural products or the pharmacophore of natural products. So only 1/3 of the compounds that have been approved as drugs really purely synthetic compounds. So it's obviously natural products play a significant role in new drug discovery. However, traditional approaches to natural product drug discovery, the one that we have used for many, many years, is no longer sufficient to provide novel drugs. So it's more and more difficult to discover new natural products. The rate of discovery of the new compound has declined significantly, and then the rate of the isolation of the same compound or known compound is increasing dramatically. So this is really a significant problem in natural product research or pharmaceutical science research in general, because it's more difficult to find new drugs. So that's called for alternative approaches, and that can be done using cutting-edge technologies and creative approaches. We can do that by looking for new sources like organisms from extreme and unique ecosystems, or symbionts or microbiomes from marine animals and so on. Also, we can use cutting-edge technologies like using new tools like new NMR technique, mass spec, LCMS, and so on and so forth. In addition, you can also use creative approaches like omics, like proteomics, metabolomics, or transcriptomics, for example, or you can use genome sequence data as tool to help you to get new natural products or new bioactive compounds that we call genome mining, or you can use biosynthetic pathway engineering and synthetic biology to discover new natural products. So my lab has been doing a lot of work in the area of natural product discovery and synthesis of natural product derivatives. Well, we use genome mining, metabolomics, and bioassay guided approach to isolate or discover new compounds. But we also use a lot of technique in biosynthetic pathway engineering and chemoenzymatic method to make new natural products. And then those compounds that we produce or tested for their biological activities. And then if something really interesting, we found something interesting, that we will study the mode of action, the mechanism, and doing some preclinical studies, mostly in collaboration with our collaborators. So today I would like to focus in one of the projects that we have been working on, and basically really focusing on one organism that can produce antibiotics, anti-cancer drugs, and other bioactive compounds, but using biotechnology or using modern technology to exploit that organism to produce as many as possible bioactive compounds. So the idea really comes from the knowledge that organisms like soil bacteria have the capacity to produce many bioactive compounds, but they are not exploited very well so far. So if you take 1 gram of soil, then you will find a lot of bacteria in there, perhaps about 10 to the 10th. This is like this is 10 billion bacteria in there. And then it has been known that, or it has been predicted that only less than 1% of those bacteria can be cultured in the lab under laboratory condition. But then even if you take, that's still a lot of bacteria. Even if you take 1 bacteria out of it and then grow it and then sequence the genome of the bacteria, then so far we have known that for every one of the bacteria from soil, they have more than 30 biosynthetic gene cluster or cluster of genes that are responsible to make bioactive compound in their genome. So basically, for every one of bacteria can make many, many different type of natural products. But in reality is under laboratory conditions, only a few of them can be identified. So many of those compounds are not produced by the bacteria under the lab condition. So the work that we have been working on really to exploit one single bacteria, but then try to make as many compounds as possible from the bacteria by doing genetic engineering, doing mutation and enzymology as well to produce compounds. So, for example, Streptomyces pecten. This is a soil bacteria that produce very famous compound called pactamycin. This compound has a very strong antimicrobial activity against gram-positive and gram-negative bacteria, but also has antifungal activity, anti-malarial activity, and also has a very good or very strong cytotoxicity. So pretty much kill all kind of organisms from prokaryotic and eukaryotic groups. So obviously this is not a a good compound as drug because it's very toxic. It was discovered by Upjohn Company many years ago, but modification or improvement of the molecule is very difficult to do because of the complexity of this molecule. So we have been working on this compound, basically trying to understand how this compound is made by the bacteria, and then try to modify or manipulate the bacteria to produce derivatives of pactamycin. So when we sequence the genome of this bacteria, we found the genome size is about 7.8 mega base pairs, and it contains 31 putative biosynthetic gene cluster, basically cluster of genes that can make different natural product, 31 of them. But in fact, in the lab, we only able to produce 4 different major products, different compound from this bacteria. So we spend a lot of time really to understand how this compound, unique cyclopentyl compounds, is produced by the bacteria by looking at the biosynthetic pathway of this compound. Not looking at, but studying it, because we have to look at the genes and the proteins or enzyme that catalyze the reaction in the bacteria to make this very complex molecule from very simple precursor or starter units. So this circle or ovals here represent proteins or enzyme that catalyze the bioconversion of this molecule step by step. So it requires so many different enzymes to make these complex molecules. We have published a number of papers reporting the process of making this complex molecule. One of the very interesting aspects of this is that the compound, this intermediate, is glycosylated. You know, the attachment of sugar, why this intermediate still attached to the carrier protein. So this protein-bound substrate that being glycosylated. And then this glycosyl moiety or sugar moiety also being modified further to make this modified glycosyl molecule. Again, while this is still attached to the protein. So this is unprecedented. It was not observed before, but this system really is able to do this. So of course, we can study this very carefully by making or producing the protein, the enzyme. This is a recombinant enzyme that we produce, and then reconstitute them or put them together back, basically taking this protein and then activate it with this phosphopentadienyl arm that can take the substrate, these 2 different substrate, and then this enzyme can catalyze the coupling reaction of these 2 substrate to make this molecule. And this molecule is then glycosylated to make this product. So this all is analyzed using high resolution mass spectrometry that we were able to confirm all this biotransformation to make the final product. This has been published about a couple of years ago, maybe 3 years ago now. And again, as I mentioned just now, the glycosylated product is being modified further. This hydroxy group is oxidized to ketone. Ketone is transferred to amine, and then the amine is carboylated to make this urea moiety, and then deacetylation occur at the end. This all while this molecule is still attached to the protein. So this is a very difficult experiment to do, but using biotechnology, we are able to confirm all this transformation. And the sugar moiety that attached to this molecule is actually the precursor of this 5-member ring or cyclopentyl unit of pactamycin, the final products. And of course, this requires further modification like methylation at this position, and then this methylation here, and then the attachment of this ring 6-methyl salicylic acid to make the final products. So these require enzymes that catalyze the reaction. And interestingly, this enzyme, this is the one that I just showed you, actually some of these enzymes, they are promiscuous, meaning they are flexible. Like this enzyme that's supposed to work here can also modify this molecule to make different compound. So in addition to this natural pathway, if you block this pathway, actually you can make a different molecule in the mutant. So that's make it possible to make or to generate new natural products that are not produced by the wild type or the natural strains. Using this biosynthetic pathway engineering, we were able to produce many analogs or derivatives of pactamycin. So this figure just to show how we can block the pathway and then make the pathway to detour to make different products. All right, some of them even we can introduce fluorine group or atom to the compound as well. We tested some of this compound against Plasmodium falciparum, the malarial parasite, the one that is resistant to chloroquine, as well as the one that is sensitive to chloroquine. As you can see, resistant strain, they die very quickly in lower concentration of chloroquine. And then this is the resistant one require higher concentration of chloroquine to be able to kill them. But our compound can kill both sensitive and resistant strain at the same time. So again, the compounds are very good anti-plasmodial activity or has anti-malarial activities. But if we look at the antibacterial activity, it's completely different story. So pactamycin, this is the parent compound, as I mentioned just now, has very good antibacterial activity, as you can see from the inhibition zone here. So against Staph aureus, against Bacillus, against Pseudomonas aeruginosa, as well as against Escherichia coli, they're all very active. But the new compound that we produce, they do not kill bacteria anymore. So now, this is good because the parent compound has no selectivities, but our new compound is selectively kill malarial parasite, but they don't kill bacteria, which is good. And then looking at the cytotoxicity as well, the parent compound has IC50 around 100 nanomolar, and the new compound that we made has IC50 1,000 nanomolar or 3,000 nanomolar. So it's about 10 to 30 times less cytotoxic compared to the natural product pactamycin. So this is just the summary of that study. The parent compound pactamycin has strong antibacterial activity, strong antimalarial activity, and very toxic. But the compounds that we modified slightly, which cannot be done using chemical approach, but we are doing this using biosynthetic approach. This compound diminish antibacterial activity, but still retain antimalarial activity and 30 times less toxic compared to the parent compounds. So pactamycin that we have studied actually is produced in a pretty low yield. In fact, in this strain of bacteria, Streptomyces pactam, produce other compounds, like I said, 4 different class of compound. One of them is NFAT133, which is produced in high yield. NFAT133 is known to have anti-inflammatory activity, anti-diabetic, and anti-protozoal activity. Then we have conlubatin here, which has anti-tumor activity, and pyrisidins that are electron transport inhibitors. Now, since NFAT133 is produced in high yield, then we also look at the biosynthesis of this compound. This is the genes. They are very organized in a very complex ways. And the way how bacteria make this molecule also very complex, right? So you can see from the starter unit here until the end. So it's require a lot of chemical reaction to make the final products. But having the knowledge how these compounds are made in the bacteria, we are able to manipulate the system and making knockout mutants that can produce new derivatives or new compounds that are typically not produced by the bacteria. So we are able to isolate all kind of different NFET133 derivatives using this approach. So in addition to that, we also can use genetic engineering really to modulate the production yield of some of the compound. So this is the wild type, the natural strains produce a lot of NFET133, produce some conglobectin, but less intense, and then pyrosidin here, pretty small amount. We can manipulate the production of this bacteria, these compounds by removing the competitor. So, for example, if we remove the genes that make this compound, so basically the mutant is no longer making this molecule, then we can increase the production of the other group of compounds like conglobectin and pyrosidin. Also, we can also knock out further the pyrosidin production, and you can increase the production of conglobectin up to like 3-fold compared to the other strains. And the same thing if you knock out the conglobectin cluster as well as the NFET cluster and pactamycin cluster, then you will be able to produce a lot of pyrosidin. This is about 7-fold increased production, because many times when we work with bacteria, the production of the secondary metabolites or the bioactive compounds is very low, which make it difficult to work on them. But by doing this genetic engineering, we can increase the production of certain type of molecules. Sometimes we can even increase the production of very minor products and then isolate them. So that way we're able to identify new derivatives or new analogs of conglobectins. And as you can see here, they have different functional group or different backbone. So conglobectin is a dimeric macrolide dialactone. So this compound is very good anti-tumor activity, tumor compound. It binds to heat shock protein 90, inhibits cell proliferation. The study has been done in HER2 breast cancer cell lines. So by identifying more conglobectin derivatives, then we can really do structure activity relationship study or to find new derivatives that have better biological activities. So we tested all the new compounds that we identified, against a number of cancer cell lines like HeLa, NCIH460, MCF7, and this murin raw 264.7 cell lines. And as you can see here, these are the new compounds. They are either comparable to conglobectin, but even some, like F2 here are more active than conglobectins, again, HeLa and NCIH460. So this is another example of making new analogs or isolating new analogs that typically you don't see when you grow the wild type. And we also tested the the dose-dependent manner of this compound. Indeed, it has or inhibit HeLa cell in a dose-dependent manner. So again, as I mentioned just now, the compounds really inhibit HSP90 especially bind to the ATP binding pocket and disrupt the complex of HSP90 and CDC37, and that's make it, resulting in dissociation of the complex with the client proteins, make the client protein easy to degrade by the system. So through this study also, we found, well, interestingly, we found very unique molecule, which is a hybrid of NFET133 on one side, and then a half structure of conlobatin. This is very interesting because these two compounds, they are derived from completely two different pathways, but somehow the bacteria can produce this kind of hybrid molecule. So just as a reference here, so conlobatin is a dimer molecule. It's made through a very complex system as well. It's called polyketide synthase pathway. Basically, the pathway make a monomer first, and then this thioesterase domain can catalyze the cyclization or the coupling reaction to make this dimer molecule. So we propose perhaps during that process, when the thioester is still holding one monomer here, NFET133 from the different pathway somehow interrupt or interfere with the biosynthesis and attack this carbonyl to make a hybrid compound. Quite interesting phenomenon. And also similarly, maybe during the process of dimerization, somehow NFET133 also can attack this intermediate to make this type of molecules. So to test this hypothesis, then we look at the enzyme. So basically we thought the enzyme can do, the thioesterase can do two different reactions. One is dimerization of this monomer to make a conlobatin, which is a dimer. But the other one, the same thioesterase may catalyze a hybridization reaction, basically using NFET133 to attack to this carbonyl to make this hybrid compounds, the same way with this one as well. So we decided to look at the activity of thioesterase. What we do is then we took conlobatin and then hydrolyze this conlobatin to get the monomer here. So the monomer, then we modified it just to mimic the way how this molecule attached to the acyl carrier proteins in the bacteria. And then this compound then is incubated with the enzyme, the thioesterase enzyme that we produce recombinantly, then together with NFET133, and indeed this compound can react with this to get this molecule. So if we use boiled thioesterase, the one that has been destroyed, this reaction doesn't occur. That shows that the enzyme really responsible for the coupling reaction to make this molecule. And interestingly, if we replace this NFET133 with other nucleophile with hydroxy group here, or even with amino group, and this nucleophile can also react with this carbonyl to make analogs or derivatives of TM127 with different side chains. So this is another example, or an example of using chemoenzymatic approach to make new bioactive compound or new analogs of natural products. So we have done this several times using different enzymes. So this is another benefit of studying biosynthesis of natural products, because you know or you understand how natural products are made in nature, Of course, they use a lot of enzyme to make them, and those enzymes can be utilized, can be used to make different compound as well, if the enzyme has some flexibility, or if they are promiscuous, meaning they can take different substrate like this. So as I mentioned just now, the production of pactamycin is very low. Because of the compound is very important. So we thought, how can we improve the production yield of pactamycin? Using the same approach, we knock out the gene cluster that make pyrosidin, so no more pyrosidin. We knock out the genes that make conglobin, so no more conglobin. Also NFET133. So this mutant where NFET133, conglobin, and pyrosidin genes have been deleted, actually produce no more of this major compound, but starting produce smaller or minor compound in higher yield. So this is just to show the genomic or generic proof of the mutant. So you see this, the wild type, and then this is the mutant. So the length of the DNA, the PCR product is smaller, meaning the gene has been deleted. So this also, this for this gene and conglobin and pyrosidin, you see all the difference between the wild type and the mutants, proving that this mutant really is not able to produce NFET133, conglobin, and pyrosidin anymore, as shown also in this HPLC chromatograms. And we also can delete all of these 4 major products, right? We can remove pactamycin, conglobin, NFET133, and pyrosidin. The mutant in green here basically don't produce them anymore, but then you start seeing other compounds being produced. So this perhaps belongs to the, the other, because in total we have 31 biosynthetic gene clusters. 4 of them has been knocked out. So maybe other gene cluster start making this compound in higher yield, of course, so that we can see them. So again, just to show all this mutant actually has a shorter PCR products because of the deletion of the DNA. So we can analyze those mutants using metabolomics analysis. This is the molecular networking or molecular network to compare the metabolites or metabolomes of the wild type, which is in red here, the triple mutants, which is in blue, and quadruple mutants in green. Blank, this is just methanol, because methanol also gives some peaks in the LCMSMS, and then the medium. So we want to exclude them from our analysis. And as you can see here, the red is a wild type. So compounds like pyrosidin or conglobin, of course, they are produced in the wild type. You can see them, but they are not produced in the blue or yellow anymore. Pactamycin produced in the wild type and also produced in the triple mutant because the gene is still there. And then you also can see in the triple mutant, the production is 3 times more than the wild type. And more interestingly is that we found some metabolites, which we don't know what they are, but they are only produced in the quadruple mutants, meaning the mutants that no longer produce the 4 major compounds, and they start producing new compounds that are not found in the wild type as well as in the triple mutant. So this is really this opens up a new area of research, try to find them or to identify those new compounds that are typically not produced by the wild type. So if you look at the antibacterial activity of the extract, then you are amazed. We are really surprised, but also happy to see the quadruple mutant. Even though we knock out all the major products, still has very strong antibacterial activities, meaning other compounds that are produced in this mutant have very good antibacterial activity, sometimes even better than the wild type, because it's being produced more than the wild type and the triple mutant. So this again support the notion that by cleaning up, some of these major compounds, then we can maybe induce or trigger the production of minor compounds, so that we can identify them and characterize their structures. So with that, in summary, as I mentioned earlier, natural products continue to be an important source of new drug leads. But again, traditional approaches no longer productive. We really need to do some other things, including using technology to improve the way how we work on natural product discovery. And then, of course, biotechnology, as I described, and genetic engineering approaches can be used to produce novel bioactive compound and increase the production of new bioactive compound as well. And then genome mining, obviously a powerful method to identify new compounds that have not been isolated before. We can use genetic genome sequence data as a reference, looking for potential new compounds. And then we also can use enzymes that are promiscuous to do modification of natural product, as well as to make new derivatives of natural products. So with that, I would like to thank the people who did the work in these projects and our collaborators here. And then this project has been funded by NIH, National Institute of Health, NSF, National Science Foundation, and USDA, Department of Agriculture. And finally, I would like to thank you all for your kind attention. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Prof. Taiful Mahmud, for informative and interesting presentation. Ladies and gentlemen, now we come to question and answer session. Now we have one question, maybe from Mr. Edwin. You hear? Okay, thank you.
Yes. Okay, good morning, Professor. I am Edwin from Faculty of Pharmacy, Pharmacy Study Program, Mandala Waluya University. I would like to ask you about the research, since like we know in Indonesia, the pharmacy student, like we are more focused on compounding and dispersing, and also like the pharmaceutical care and something like that. But we rarely do the research on this kind of research, like biological research using the genome and something like that. And I would like to ask you, in your opinion, like how far we should learn, how far we should study about the biological engineering, biological process, like your research, like biosynthesis and genome editing and something like that, since it is really rare in Indonesia. But I think this kind of research nowadays is really popular among the researchers around the world to make or to produce the new therapy for disease. Thank you.
Right. Thank you very much, Pak Erwin. Can you hear me? So, well, it's of course depend on what you want to achieve, right? I mean, if you are interested in doing more cutting edge research, and then in the drug discovery arena, I think we really have to broaden our research portfolio, including using genetic and metabolomics or proteomics approach, because those approaches are very powerful. And then, of course, compounding is also important. In pharmaceutical sciences, of course, you have at least 3 different big division, right? I mean, pharmacology, pharmaceutics, and medicinal chemistry, or drug discovery. So if you are focusing on pharmaceutics, and of course, compounding is one of the aspect there. But again, even in the area of pharmaceutics, nowadays, traditional compounding is no longer interesting, I guess, because people already moved to the nanotechnology, drug delivery system, and liposome and all those stuff that to catch up with the advance of science and to compete with other scientists in the world. Then obviously we need to also go to that direction. And in the case of natural products discovery, for example, or even in the case of say vaccine and enzyme and so on. So there are a lot of genetics and molecular biology technique that we need to learn before we can even do anything in that area. So I think as a pharmacy education or institution, I think we really need to incorporate all those new technique, at least to familiarize our student so that they know actually how this RNA vaccine is made, and can we do the same, or a protein vaccine, or or modifying bacteria to make new compounds. Because if you only rely on natural sources like plants and bacteria and fungi, and then try to isolate compounds from them, it is more and more difficult nowadays, really, to discover anything new. So for that reason, I think it is time to start something new, and I mean, it's to be more competitive, I guess. Thank you. I hope that's answered your question.
I have some question too, if you don't mind. Sometimes ago, I did research on anti-diabetic mellitus. Interestingly, in your presentation in NATT 130T synthesis, can be used as anti-diabetic mellitus too, right?
Sorry, can you speak louder? I cannot hear you very well.
Okay. Sometimes ago, I did research on anti-diabetic mellitus. In your presentation too, in NATT 130 synthesis can be used as anti-diabetic mellitus too. Right?
Oh, I see that the NFAT 133.
Yes. To develop
So,
this discovery, what step we do need to take?
So yeah, so that compound has been reported to have anti-diabetic activities. But well, we didn't do the experiment. It was reported by somebody else, and it seems like it has not been, no further study has been reported. So I don't know how far they have been they have done, the study, and we don't know whether the compound can be used as anti-diabetes in humans, because knowing that the compound has several other biological activity, including inhibiting a nuclear factor of T cell. So yeah, obviously more study need to be done to know whether the compound is really useful for diabetes.
Okay, thank you very much, Professor Taiful Mahmud, for your answer.
You're welcome.
Now, you have any question here? Good. Mr. Yusuf Hussein. Question.
Ladies and gentlemen, good morning for everyone. Good morning, Prof. Taiful Mahmud.
Good morning.
My name is Yusuf Hussein. I am Secretary of Mandala Waluya Foundation, the organizing body of Mandala Waluya University. I want to ask you one only question. Have you come to Indonesia?
Yes, but Yusuf, I'm Indonesian.
Oh,
So
you're Indonesian.
yes, so I was born in Aceh,
Oh,
and yeah, I got my bachelor degree in apothecary. I'm apotheker from University of North Sumatra, USU.
thank you very much. And I am sorry because my English is not so good. Thank you.
That's fine. That's fine. Thank you.
Okay, thank you, Mr. Yusuf Hussein from University of Mandala Waluya, Kendari. Now, any question, or we pass to the next session? The next session is the virtual certificate. Let me representing this committee to give you virtual certificate. Are you ready, Professor Taiful?
Yes, thank you.
Okay. Can we take a picture together?
Okay, thank you for the moderator, and to Professor Taiful Mahmud. So we would like to ask your sincerity to take a picture together with our committee, also our moderator, and for the participant. Okay, for the committee, please to take a picture of the screen. Done. Okay, thank you. I give back to Miss Rifa to Mahmoud. Yeah.
Okay, thank you, DMC. Now, yeah, thank you, Professor Taiful, for your time to join this conference. Now
You're welcome. Thank you.
Now we can move to the next session with Professor Muhtaridi, PhD. Are you here?
Yes, I'm here.
Okay, we hear you. Professor Muhtaridi, as we know, the CV I have been read before. Now, allow me to invite the Professor Muhtaridi to for presentation, the material of presentation. Professor Muhtaridi, time is yours.
Okay. Thank you for Chairwoman. Let me share my slide first. Okay. Okay. First of all, I would like to thanks for kind inviting me, especially Dr. Yuli, head of program of pharmacy, yeah, Universitas Mandala Waluya. Of course, I'm very happy to join this seminar with you. Also, thanks to Rector of Universitas Mandala Waluya for the facilitate this seminar, Dr. Ratna Umi Nurlila, MSc. As mentioned, Chairwoman, I'm coming from Universitas Pajajaran. Actually, it should be located in Bandung, West Java. However, we have office in Jatinangor, Sumedang. Yeah. Some lecturer from Mandala Waluya also is taken PhD from our university. Okay. Today, I would like to take this opportunity to present about, as you can see in the slide, drug discovery, radiopharmaceutical from natural compound in Indonesia. Yeah. So I'm from research collaboration for radiopharmaceutical theranostics brain also, because we have project big grant project from with this. Yeah. Okay. Why we take this topic? This topic was taken due to the trend of radiopharmaceutical is increasing because of its use in hospital, which is very helpful in detecting disease and treating cancer. On this topic, I will explain how the potential for radiopharmaceutical development in Indonesia, and it's up to our experience in grading of radiopharmaceutical. Okay, let me introduce what kind of radiopharmaceutical. Okay, radiopharmaceutical is defined by Saha is a radioactive drug used for diagnosis or therapy in tracer quantities with no pharmacological effect. So the chemical compound or drug of radiopharmaceutical is which one of the atoms making up the structure is radioactive nuclide. For the purposes of diagnosis and therapy against disease like the cancer, tuberculosis, and so on. Okay. Use of radiopharmaceutical and radiochemical principle and methods to make drug containing radioactive or radionuclide atoms for the purposes diagnosis and therapy disease. Some radiopharmaceutical terms that should be known, like the radiopharmaceutical is used for nuclear medicine and 90% for diagnosis. You know that no minimum pharmacological minimum pharmacological effect. For therapy can cause tissue damage by radiation, and the radiopharmaceutical is sterile and pyrogen-free and complies with quality control like the conventional drugs. Okay. In radioactive such as iodine or labeled iodine protein, labeled compound, 19 technetium is the almost radiopharmaceutical label. Yeah. Other term for radiopharmaceutical is the radiotracer, radiodiagnosis agent. Okay. In general, these are the radioisotopes that have been used in radiopharmaceuticals and supporting instrument for the application like this, medical imaging techniques such as positron emission tomography and single photon emission computed tomography spec rely on cyclotron produced radioisotope and so on. However, unlike reactors, which also produce radioisotope, cyclotron this one, do not use nuclear materials and are not subject to some radiological safety and security consideration as reactors. Okay, radiopharmaceutical doses, are administered into the patient in patient's body with a ligand carrier that can bind the target. Okay, the radionuclide labeled on the ligand pair emits radiation at the desired target, and computer system read the emission through a scanning device and then analyzed by a nuclear medicine specialist in the hospital. Okay, here we resume the target of the particle emission alpha, beta, and gamma. Alpha, gamma is long radiation, right? So gamma emission is for diagnosis, alpha, beta for the therapy. Okay, here we resume long journey in radiopharmaceutical development in the world. Brugarolas et al. Summarized the Journal of Radiopharmaceutical in their article entitled 15 Years of Radiopharmaceuticals. And here you can see that bismuth and technetium, phosphor, and natrium chloride radiopharmaceutical radioisotope were discovered in 1920 until 1913. And then FDA started announce the using the regulation radiopharmaceutical. Also, FDG for fluor deoxyglucose for metabolism was introduced in 1970. In 1980, FDA many approved some radiopharmaceutical, and first one is technetium hexamethazine for diagnosis of stroke. Okay, also FDA approval rubidium active 2 for positron emission tomography, myocardial perfusion imaging. In 2000, positron emission tomography and CT scan was developed to use to supporting radiopharmaceutical in hospital. Okay. In 2010, FDA approved many radiopharmaceutical. And 2020, the radioactive radioisotope for the radiopharmaceutical is many launched to expanding the chemistry toolbox for the synthesis and new radiopharmaceutical. So now radiopharmaceutical expand and explore wider access, particularly in developing nation, including Indonesia. How about Indonesia? So I explain about Indonesia here is distribute of radiopharmaceutical facility in Indonesia. In hospital in Indonesia that provide nuclear medicine facility. Okay. This one you can see the hospital in Indonesia that has nuclear medicine. And in the future, these facilities will be stand in 13 provinces of Indonesia. This is component of nuclear medicine facilities, which are owned by 8 hospitals, including SPEC, public hospital, cyclotron, and so on. Okay. Current condition, Indonesia has 12 hospitals that provide nuclear medicine facilities. Yeah, Indonesia only has 4 radiopharmaceutical centers of production that produce radiopharmaceutical cyclotron. The first Rumah Sakit Gading Pluit, MSTC Siloam, Cancer Darmais, Abdul Wahab Samarinda. These collaborate within research institution BRIN, yeah, universities like Universitas Pajajaran, and hospital to support development and radiopharmaceutical Indonesia. This roadmap of radiopharmaceutical Indonesia, okay, we classified the research by its technology readiness level. In Indonesia, we call tingkat kesiapterapan teknologi. Okay, here we TLL2, curcumin gallium, curcumin labeled iodine, and TLL3 alpha mangostin labeled technetium, alpha mangostin labeled gallium, and so on. And this our product. Yeah, our product is alpha mangostin, iodine 131, quercetin 131, technetium quercetin, and curcumin scandium. Okay. Okay, this is the antibiotic labeled kanamycin and ketoconazole produced by BRIN, yeah, by BRIN or obatan, yeah, the past year. Okay, this the chemical compounds synthesized we produce in Indonesia by Batan, BRIN, sorry, MA8, tetraphosphine, and nano albumin, yeah, also glutathione technetium and CTMP technetium. This peptide synthesis and molecular organic that already commercial is MIBG and ethambutol. Also, this already commercial some kits, yeah, kit MIB, kit MIB, kit MDP, and kit region, yeah, and DTPA. This one, okay. Okay. So Indonesia, you know that Indonesia is the ranking of 2 mega biodiversity of the world. So Indonesia has an abundant source of natural compound. So the natural compound have the potential in discovery of new drugs, including radiopharmaceutical. One of natural compound is alpha mangostin with activity against breast cancer. So in our university, Universitas Pajajaran, we conducted the research of radiopharmaceutical from alpha mangostin, and each was started in 2018, 2019, yeah, from labeling alpha mangostin using technetium 99M alpha mangostin, and this research is continued until now. Yeah, for the okay. So what is alpha mangostin? You know, alpha mangostin is xanthan derivatives that isolated from mangostin pericarps that has pharmacological effect like the anti-cancer, cardioprotective, anti-inflammatory, anti-acne, anti-tuberculosis, antioxidant, antibacterial, antifungal. So alpha mangostin again breast cancer based on in silico study we conducted using molecular dynamic, pharmacophore, free admetox, and so on. This we published in JLPTR. So this we published already accepted in later in drug design discovery Bentham Science that we studied molecular dynamic and pre-admetox of alpha mangostin. Then we know that the alpha mangostin has potential as the anti-breast cancer. The in vitro study of alpha mangostin produced IC50 value 0.044 microgram per milliliter. It's more prospect than tamoxifen 0.1 microgram per milliliter. So we can say that alpha mangostin is potential for the anti-breast cancer. Based on in silico, we in silico and in vitro, we assay in vivo study. You can see that in pure alpha mangosteen, so it's tended to be stable in the first week and experienced a slight increase in the second week. So we compare then nano alpha mangosteen, nano amcal, at those 20 milligram, so a graphical decrease that was not much different from the tamoxifen group, and a significance value 0.036, which means that no significant difference between due to the tumor volume continuum. Based on this result in silico, in vitro, and in vivo, we label alpha mangosteen using radioisotopes in this organ. Yeah, you can see that alpha mangosteen is very good to kill the cancer cell of breast cancer. Okay, so we labeled alpha mangosteen using technetium with the scheme study. First one, optimization, radiographic condition with pH, natrium DTA, and selenium chloride. And we analyzed the radiochemical purity and identification using paper chromatography, TLC, electrophoresis, and so on. And we check the impurities, pertechnetate and technetium oxide. Okay, this is the formula of the of alpha mangosteen technetium, and this is the result graphic, the LSP analysis of the optimum condition of technetium of alpha mangosteen measured through the PPC and TLC, paper chromatography. As shown in the above figure, TLC PPC could not spread 99 M technetium alpha mangosteen and 99 M technetium EDTA. Furthermore, electrophoresis and HPLC were needed to ensure that 99 M technetium alpha mangosteen and EDTA. The electrophoresis was conducted by analyzing technetium alpha mangosteen and technetium EDTA as control. The result was radioactive migration pattern of the sample, which consequently compared to the control. The result showed that the three samples had different migration pattern. The peak of control 99 pertechnetate at the rank migration per centimeter is the peak technetium amount. Yeah, so we can spread. So this has PLC data. We can conclude that alpha mangosteen technetium not perfectly separated, yeah, by comparing migration pattern of each sample. So this failed, yeah, the research. So also we optimization the result, yeah, for the using iodine, yeah, using iodine. Okay. The radial bring alpha mangosteen with iodine. In the radio iodination, we conducted optimization of reaction time, temperature, and chloramine. Yeah, chloramine. CAT is chloramine, and alpha mangosteen to obtain formula with highest radiochemical purity. After that, we performed the in vivo and in vitro and in vivo study. In vitro study is cellular uptake, radiopharmaceutical in MCF7 and T47D. Yeah, in vivo study consists of biodistribution and pharmacokinetic and toxicity. Okay, radio iodination using chromium T as or CAT, oxidizing agent, and the mechanism of reaction electrophoresis. This CAT will react with iodine to product iodine with positive charge. The oxidized iodine will substitute hydrogen in benzene ring and produce iodine 131 alpha mangosteen. This is the mechanism of reaction. This optimization radioiodination, the optimum condition was obtained on time reaction 30 minutes, temperature 2 degrees. and CAT concentration 1 to 5 microgram, and alphamocin 312.5 microgram. Radiochemical purity was conducted by electrophoresis with formula, this one. Okay. So we have purity 900% minus percent impurities. Okay. Cellular uptake test was carried out in MCF7 and T47D breast cancer cell line with incubation time of 10, 30, and 60 minutes. The results saw that you can see that T47, the highest uptake was obtained on 30 minutes incubation. Meanwhile, in MCF7, the highest uptake was on 30 minutes incubation. After in the in vivo study, yeah, we use in vivo study, the first distribution study predict distribution radiopharmaceutical in organ, and this the result. You can see that the next study is acute. The pharmacokinetic study, so that iodine 131 alpha mosine has half times distribution 0.54 and elimination of 75.77. The next study is acute toxicity study. Is okay. It showed that alphamocin doesn't cause mortality toxic symptom in the animal. Okay, this is the result radiolabeling estradiol for the radioligand binding assay. In this study, direct synthesis estradiol using iodine 131 has been carried out using radioiodination method. This synthesis was chosen because it's relatively simple and easy for radiolabeling. Okay, so we published in the Helion journal this scheme research. Yeah, okay, I try to be fast. Okay, beta as well has been labeled, yeah, by natrium 131. Yeah. Is the radioiodination reaction. Radioiodination is method label substance using gamma emitting radioisotopes of iodine, such 125 iodine and 131 iodine. This method able to produce label compound with the hex-specific radioactivity at very low concentration. This from optimization, we can obtain formula, this one, yeah, this one. Reaction time 5 minutes and so on. So the radiochemical purity was analyzed by HPLC, the optimal separation obtained with flow rate 0.75 with azotate water. And this is the peak result. Yeah, you can see that. Okay, we got 75.84% is good result. Yeah. Okay, we also using cellular uptake test evaluation on estradiol iodine. Yeah, this one the result. Okay, and the next I try to explain the future prospective from natural compound. So we publish in Molecules by our students. Yeah, radioisotope. Okay, the natural compound is mostly distorted from plants, as mentioned Prof. Taipo Mahmud. Yeah, this natural compound are labeled with iodine radioisotope. This is the result year to year. Yeah, so we assume that this one. Yeah, so the development stakes of radiopharmaceutical from natural with iodine radioisotope are rather long and challenging. The challenges reported in previous studies were mostly related to radiochemical purity and BI distribution. Problem related to radiochemical impurity. Radiolabeled compound have low radiochemical purity, less than 95%. Yeah, is for example. Okay, this okay is the how to strategy. So we give the strategy. Radiochemical purity is crucial for crucial quality control factor in radiopharmaceutical development. The radiochemical impurities of iodine are free ion of iodine and iodine, which affect the safety and accuracy of radioparticle. So the first strategy to obtain maximum radiochemical purity is to select a suitable radioionization method based on the steric characterization. The first radioaddition electrophilic saturation, substitution, and the first method using chloramine T. The critical point is the reaction RPH. Okay. Okay, formation impurity iodine indicates low in vivo stability, yeah, in the radioparticle due to the deiodination reaction. The accumulation of free iodide as impurity in thyroid, stomach, and intestine reduces the target to background ratio of the diagnostic agent. Thus, the diagnostic results are biased, yeah. Okay, this the for example. Okay, another strategy that we applied to increase the target specificity using nanoparticle play a role increasing the penetration compound across biological, yeah, membrane, so they can effectively deliver therapeutic agent. Yeah, so we use membrane reduce. Nanoparticle can increase the deliverance specificity of radiopharmaceutical toward its target by conjugating the target molecules. Okay. This another strategy. Yeah. Okay. So the conclusion of this presentation are Indonesia has abundant source of natural compound. This natural compound have the potential in the discovery of new drug, including radiopharmaceutical. One of natural compound is alpha-mangosteen isolated from mangosteen, pericarp that not has activity against breast cancer. Due to its activity, alpha-mangosteen could be used as pharmaceutical case of radiopharmaceutical breast cancer. Radio labeling alpha-mangosteen with technetium sought the low radiochemical purity. Radio labeling alpha-mangosteen with iodine saw good radiochemical purity, and the research was continued on in vivo study. To obtain radioligand binding of radiopharmaceutical, radioionization of estradiol was conducted, and it showed good RCP and uptake MCF7 and T47D lines. In order to support this research, we brought the literature review on radiopharmaceutical from natural compound. Okay. So this our collaborator, our team of Pusat Collaborasi Riset Radiopharmaceutical. We have three professors. Professor Hussein Kata assessment from Hospital Hasan Sadikin, Prof. Udin from BRIN, and this hospital, Dr. Rian from Siloam MCC, Dr. Ayub from Darmais, Dr. Eli from Rumah Sakit Hasan Sadikin, and Pak Freddy from ITB, Pak Denny and Pak Supriyanto from University of Indonesia, Pak Holis, Pak Sandra, and Burani from Pajajaran. This is our student for radiopharmaceutical research. Yeah. So our collaborators, Prof. Tomoya Uhara and Dr. Kanaka Sensei from Shiba University, and Prof. Sarum Samsudin and Dr. Wan Azumi binti Muhammad Fuad from Universitas Malaysia for the toxicity study. Okay, thank you for to listen my presentation. So I give back to Chairwoman.
Very interesting, and you have many publication in the radiopharmaceutical natural product. Next we have question and answer session. The question from the all participants.
Okay.
Maybe
Can I have a question?
okay. Okay, maybe from Mrs. Nur Hatijah.
Thank you very much, sir, for the nice presentation. As we know, the Indonesian have much product, natural product. So I want to ask how to combine the radiopharmaceutical suitable for the natural product. Thank you.
Okay. Thank you for the question. I answer directly.
Yes, you can
Okay.
answer directly.
Okay, good question and interesting, yeah. How to choose the natural product compound can label it by radioactive and as radiopharmaceutical. The first one, we can say we can find the activity, yeah, activity on targeted. You make sure the natural compound must be targeted to organ, yeah, and selective, yeah, and selective, yeah. After we know how to know this natural compound is selective, we can use in silico, first one. You can use in vitro Western blot, yeah, reaction. You can use in vivo using the biomarker. So after that, if we know that these compounds bind with the target organ like the cardiac and so on, yeah, kidney. So we can label the compound. Can label if have the benzene ring, hydroxyl groups, and amine groups. Yeah, so we can label this compound. Okay. Hopefully satisfied for my answer. Thank you.
Okay, thank you, Professor Musaridi, for the answer question. Next, I have any question?
Thank you for the moderator. Maybe this is for the first time NMC have the question for the seminar. Okay, Professor Mohtaridi, perhaps my question is a bit far from your topic. My question is about, I just want you give us an insight because my research now currently in a bacteriophage using to recover the drug resistance. As we know now, we using drug. Some people have a drug resistance and they use for natural product. As you explained just now, even we use natural product, there is a tiny problem in a radiopharmaceutical, right, sir? So perhaps how we can solve this one if we use the natural product, but still we have the problems in front of us, like when we use metformin to cure the diabetic, but it is have a problem with the people with have the kidney disease. So perhaps you have an insight, which one is the better one? We use a drug, a chemist drug, and then we use natural product, or we use our immune system, our own immune system, by using the bacteriophage in our body. Thank you very much.
Okay. I forgot the name, but this good question, very, very good question and interesting. Yeah, your question is which one better? Yeah, using single compound or synthesized compound, natural product, or to reduce the resistance. Okay, the resistancy of the drugs if drug is single compound and from synthesized and used routinely, yeah. So our body will be now this compound, the pattern of this compound. So the like the protein will be communicate with the amino acid and the other amino acid to know that this the drug, this the inhibitors. So we can prevent of this drug. So that's why metformin, tamoxifen will be resistant into the target. So, how about the natural compound? So if you have natural compound from isolated, it's safety, but I prefer for the natural, like the natural product, not isolated compound, because natural product like the extract, simplicia is a complicated compound. Yeah, the many compound on the on the extract and the simplicia. So our body is your target, your disease is confused with the, which one the target. When they know some compound from the from extract, the other compound will be active to against of the target. So how to solve it? So we can modify the compound, like the for cases, yeah, for avian flu, when we created when the researcher created oseltamivir or Tamiflu. The first one is very good activity against avian flu, but after that, after 3 years, this drug developed, we found the resistance of this drug. So the researcher modified this oseltamivir to the other compound like the zanamivir. After that, 3 years ago, 3 years later, the zanamivir is resistant, and again like that. Yeah. So why natural product is very safety for our body to use. Yeah, but natural product cannot directly to reduce the to against the the disease. Okay, that's my answer. Thank you.
Okay, thank you, Professor, for the answer. Back to moderator.
Okay, thank you for the answer, Professor Mustari. Maybe you have the last question. Okay, please, Mr. Edwin, please the last question for Professor Muhtaridi.
Okay, good morning, Professor. I'm Edwin from the School of Pharmacy. I would like to ask you about the development of radiopharmaceuticals. Since like this kind of therapy need to be selectively targeted to the spot.
Sorry, freezing, yeah. I think freezing the host. Hello. Hello. Hello, committee. I think host of yeah. I'm also not sure. Okay.
The next session, I am returned to the
Okay, ladies and gentlemen, because our keynote speaker, Professor Apotheker Muhtaridi, has a problem with the network. So for the next question from Mr. Edwin, perhaps can send to our head of Department of Pharmacy, and Professor Muhtaridi will answer it. And for the next session is given the certificate symbolic for the Professor Muhtaridi, but because the signal is not good. So I give back to moderator.
Okay, thank you for the session. Maybe I apologize with my maybe many mistake in the session. I hope you can we meet again in the next conference. Again, I back to the MC to the next session. Okay, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Big applause for our moderator and our keynote speaker, Professor Taifo Mahmud from Oregon State University, USA, and Professor Apotheker Muhtaridi, PhD from Universitas Pajajaran, Indonesia. And now we still have like minutes to move to the next session. Is an invited speaker. We will present three invited speakers. The first speaker is Associate Professor Dr. Najia binti Muhammad Hasim from University of Malaya University. And then the second one is Dr. Suman Sinha, Institute of Pharmaceutical Research, GLA University, India. We apologize if the spelling of GLA is not the exact one. And the third one is Amirah Muhammad Ghazali, PhD, a registered pharmacist from University Science Malaysia, Malaysia. Now I would like to invite Mr. Apotheker Edwin Jacob Bungulawa as our moderator for this invited speaker session. And welcome Mr. Apotheker Edwin Jacob Bungulawa, Master of Engineering. Will be led. The time I give to moderator.
Okay, good morning, everyone. Thank you so much for the time. It's an honor for me to be your moderator right here in the second International Conference of Mandala Waluya Department of Pharmacy. I am Edwin from the School of Pharmacy also. Today, I will be your moderator for three of our invited speakers. Maybe you can show the okay, our first invited speaker is Dr. Suman Sinha. He studied in the Berhampur University to get a bachelor degree from 1997 to 2001 and obtained his Master of Science degree as a Master of Technology on Pharmaceutical Chemistry in Fuller Institute of Technology in 2004 until 2006 and continued studied the PhD degree in the Doctor of Philosophy for Pharmaceutical Technology in Medicinal Chemistry at the University of Science Malaya in 2011 until 2017. His career experience is medical representative with Nicholas Pyramidea Limited in 2002 until 2004 as a lecturer in Roorkee College of Pharmacy and Allied Science of Uttar Pradesh and is a senior lecturer now in the Department of Pharmacy at Lovely University in Punjab, India. He is also an assistant professor in the Department of Chemistry at Cairo University from 2009 until 2011 in Tamil Nadu, India. Also research assistant of Department of Pharmaceutical Technology, School of Pharmaceutical Science, University Science Malaya from 2011 until 2017, and currently postdoctoral fellow in Tata Institute of Fundamental Research in Hyderabad since 2017 until now. He published several papers in Indonesia and currently is honorary professor. He published papers focusing on medicinal chemistry, computer-aided drug design, and organic chemistry, and participated in hand-on training and different techniques in drug discovery with Origin Discovery Technologies in India also from August to September 2013, and participated in short-term research project on molecular dynamic simulation as a visiting student with Dr. Jagannath Mondal at Tata Institute on Fundamental Research Center for Interdisciplinary Science in India from August to September 2016. And currently he is a researcher in the development of a competitive protein inhibitor against therapeutic targets in malaria infection and the development of peptide inhibition against AMI RON2 interaction and computational structure biology using atomic molecular simulation. And he has been applying both equilibrium dynamic as well as enhanced sampling techniques to understand free energy surfaces pertaining to molecular recognition and elucidate also. Okay, we move to our second invited speaker is Dr. Amirah Moh Ghazali, PhD RPH. She studied in University Science Malaya, Penang, Malaysia for bachelor degree of pharmacy and graduated with the CGPA 3.57. She's obtained like Master of Science in the University Science Malaya also in Penang in 2012 and PhD degree in the University of Lorraine, Nancy in France in 2016. And you can read in our slides. She also have many experience such as in May 2007 until May 2008, the research exercise elective paper in pharmacy course, docking simulation, study of 200 compound derived from Malaysian common plants on vascular endothelial growth factor receptor 2 or VEGFR2 in 2010 until 2012, Master of Science Pharmacy, University Science Malaysia in formulation and characterization of rifampicin loaded with 3-HB co-4-HB nanoparticles for oral anti-tuberculosis therapy. And then in 2013 until 2016, taking his doctorate of science in the University de Lorraine, Nancy, France, designed the targeted photosynthesizer conjugates toward neovilin and folate receptors for improving the selectivity of anti-cancer photodynamic therapy. And last but not least, the Associate Professor Dr. Najiha Moh Hashim. Graduated with Bachelor of Pharmacy from the University Putra Malaysia, Penang, and obtained her postgraduate degree in Natural Product Chemistry from the University Putra Malaysia. Since her appointment at University she has held several important posts at the department, faculty, and university and national level, including academic and research, as well as professional service. She's currently Deputy Dean of the Academic and Faculty of Pharmacy, and the head of CENAR, or Center for Natural Product Research and Drug Discovery. Okay, you can read the rest on the slide.
Excuse me, for the moderator. For the first session is we have Associate Professor Dr. Najira as the first speaker for this invited.
Okay.
And the second one is Amirah Muhammad Ghazali. Thank you, sir. And the time is 25 minutes for
Okay.
each. Yeah, thank you.
So for the first time is Associate Professor Dr. Najiha Moh Hashim, right? Good morning, Dr. Najiha Moh Hashim. Are you with us? Can you hear me? Good morning. Dr. Najiha, can you join with us now?
Perhaps your sound is a little bit louder, so Professor Dr. Najira can hear you in a screen, because the voice in a Zoom is so lower.
Okay. Dr. Najiha, are you with us now? Can you join the seminar? Okay, then we invite Dr. Amirah Moh Ghazali, PhD RPH, Dr. Amira, can you hear me? Dr. Amira, could you please join the seminar? Okay, I will call again Dr. Najiha for or Dr. Amira. Doc, Dr. Amira, can you hear me?
Yes, Assalamualaikum. Can you hear me?
Yes, I can hear you. Okay, thank you so much for your time. How are you?
Good. Thank you for the invitation. Thank you very much.
Okay, you're welcome. Welcome to our second international conference. Okay. You can start your presentation in 25 minutes and we will back to you on the question and answer section. So the time is yours.
Okay, thank you. So let me share my screen. Yeah. Can you see my slide?
Yes, we can see.
Okay. Okay. So a very good morning Mr. Chairman, dearest professors, doctors, Bapak-Bapak, Ibu-Ibu, postgraduate students, and dear participants of the second Mandala Waluya-International Conference on Pharmaceutical Sciences and Practice. First of all, thank you very much to the organizing committee for inviting me to share some insight and thoughts on the current technology in pharmaceutical science research. So my name is Amira, and I am currently at the School of Pharmaceutical Sciences, University Science, Malaysia. So we are located at the north of Malaysia at this small island, Pulau Pinang, or also known as Penang. So our campus is also known as a University in the Garden. So I would like to extend the invitation to everyone if you have the opportunity to come and visit us in this small and beautiful island. So my talk today will focus on the application of advanced delivery system in the development of targeted drug delivery towards tuberculosis or Mycobacterium tuberculosis. So the title of my talk will be War Against Tuberculosis, Modifying the Drug Delivery Approach. So before we move further, let us take a look or revise, okay, what we know about TB or tuberculosis. So tuberculosis is caused by a bacteria, Mycobacterium tuberculosis. So mainly it will infect the lung. Okay, this is a photo, okay, of the TB Mycobacterium. So usually it will invade the lung, but it can also invade other organs. So it can invade the spleen, the joints, okay, causing extrapulmonary tuberculosis. So the number of cases worldwide actually is quite worrying. It's about 10 million per year, okay, and the number of deaths can reach about 1.5 million per year. So this is a huge number because, as we know, tuberculosis is an infectious disease that can be treated with antibiotics. But with the presence of effective antibiotics, this disease is still difficult to manage. Why? Because there's some issues with the treatment. As an example, the long treatment duration. So this will affect the compliance of the patient. So these are some newspaper cuttings from the news in Malaysia. So ever since the beginning of pandemic COVID-19, in the recent years, it was clearly observed, okay, that the number of cases for TB has been increasing. Yes.
Your slide is not changing in our screen.
Oh, wait. My slide is not changing. Wait, okay. Can you see?
Okay, yes.
Yeah.
You can see it now.
Okay.
Thank you.
Thank you. Sorry, So the number of cases has been increasing. Why? Because when patients were infected by COVID-19, the immune system reduced, right? So when this happens, it can lead to the infection by more dangerous organisms such as Mycobacterium tuberculosis. So I have a personal experience whereby my own sister was infected by TB after she was infected with COVID-19. And now she is under treatment, under the 6 months treatment with the 4 antimicrobial. So it's like it's not very common, but it started, the number of cases has been seen increasing. And this also involves TB in children. So we have to be aware on this current situation. So, okay. So according to the WHO Global TB Report 2021, 43% of the world TB burden is in the Southeast Asia. This is where we are located, right? So in Malaysia, in Indonesia, Thailand, Singapore. So these are the countries that has the 43% of the world TB burden. So this tell us something that we have to be careful and we have to do something to contribute to overcoming these issues, and also how we can meet the target set by the United Nations through the Sustainable Development Goals. So if we look at the SDG number 3, the good health and well-being, there are 2 targets specifically made for infectious disease such as TB and malaria. So target 3.3 is to end the epidemics of TB by 2030. And target 3B is to support research and development of vaccine and medicine for TB and malaria, and also AIDS in this case. Okay. So the WHO has also put up the consolidated guidelines on TB for prevention, screening, treatment. Okay. So if needed, you can always refer to these guidelines. So going back to the treatment, okay, the current anti-TB treatment available. Currently, we know that for anti-tuberculosis treatment, we have four main drugs that are being used, right? The first line, rifampicin, isoniazid, pyrazidamide, and ethambutol. The four combination has to be taken for a minimum of 6 months, 2 months for the 4 drugs, and the other 4 for the 2 drugs. However, there are some issues. Okay, as an example, when these drugs were launched, okay, more than 50 years ago, during that time, the research for the pharmacokinetics, pharmacodynamics of the drug is not as extensive as what we have nowadays. So when the drug was taken orally, right, and then the drug molecule in the blood vessels, the drug molecule has to penetrate through the blood vessels into the granuloma. This is an example of granuloma presence in the lung. Okay, for it to penetrate, it has to go through the different cells available here. We have macrophages, we have foamy macrophages, okay? They have to penetrate. So this penetration is not as simple, right? There are so many layers. So this is the reason why the certain concentration of anti-TB drug has to be remain or has to be sustained in the blood. So if patients is not compliant to treatment, this will lead to reduce blood concentration of the drug and subsequently resistance of TB. So penetration is not difficult. So penetration is challenging, okay, and it can be it has to be reached, okay. The drug has to reach the center of the granuloma in order to eliminate the Mycobacterium tuberculosis. So this is one of the challenges. So this bring us to the detail, okay, especially on the ADME of the 4 drugs. Rifampicin is known to have low solubility, okay, and pyrazidamide, as an example, is known to have some permeability issues. So these are the problems, okay, that may affect the effectiveness of the treatment. Although we know the treatment is effective, but the duration is long and the dose sometimes has to be up, okay, so that we can reach the targeted concentration in the blood. Okay, so what is advanced delivery system? So if we can imagine, okay, like in this picture, okay, if we can have a smart delivery system, a small molecule with that is programmable. We can program it with where to go and what to kill. So it is nice, right? So when we have a targeted delivery system like this, we can target the drug to the diseased tissues. Okay, so it is specific, and we can subsequently reduce the dose and minimize the side effect. So this is what targeted drug delivery system aim. Okay, so to have a specific delivery and hence to concentrate the drug concentration, okay, specifically at the cells, okay, at the disease cells. Okay, so if we look at the concept of delivery system, according to the team, okay, to the paper published by Tiwabe et al. In 2021, there are 5 generations of drug delivery system, or DDS. The first generation is the conventional form that we have commonly in the clinical setting. We have capsules, tablets, right? And the second generation is more like the modified action system, such as the enteric coating, tablets, right? So this is the second generation. The third generation is more on the control delivery system, when we can control the rate of the delivery. And the fourth and the fifth generation is where the targeted delivery is. The fourth generation is when we target specific to the disease cells, such as in the nanoparticulate delivery system. And the fifth generation, the most advanced that we have now, is the gene therapy and biologics. So these drugs such as teratizumab, etc., monoclonal antibodies, right? They can modify our immune system to help in the treatment of certain diseases. Okay, so the concept of targeted delivery, drug delivery actually aims to reduce systemic drug toxicity. Why? Because when we target certain cells, as an example, if we can target the macrophages that surround or that or engulf the Mycobacterium tuberculosis, we can ensure the delivery is specific to the Mycobacterium tuberculosis and not to the other part of the body, right? So, so this is what we aim to do with targeted delivery system. So among the studies that being conducted is by producing nanoparticulate delivery system as liposome, polymeric nanoparticles, okay, at the nanoscale or the micro scales. And when these are engulfed by the macrophage, okay, it will release the encapsulated anti-tuberculosis drug. So this is the aim, okay. So in targeted drug delivery, we have two approaches. We have the active delivery and also the passive delivery. So in the passive delivery, okay, I begin with the passive first. Passive means we use the nanoparticles as an example. So the delivery will more like due to the macrophage engulf or due to the penetration through the blood vessels, etc. It's more like passive flow. But for active targeting, we use a specific ligand that we attach to the drug that will help to bring the drug specific to the targeted or to the disease cells. So this is more active approach. Okay, so how can advanced delivery system helps? So as an example, if we can deliver the drug specifically to the target, we can reduce the dose and we can reduce the possible side effects. Because one of the reasons for the multiple side effects experienced by TB patients is due to the high dose of the drug use. So, and also we can release the encapsulated drug via physiological or chemical trigger. So as an example, we formulate a nanoparticle system. When it's being engulfed by the macrophage, there will be changes in the pH, in the surrounding pH. So that's when the drug will be released from the nanoparticle. So this is one of the way, okay? So we can localize the delivery only in the macrophage. So that is one example. So this is a study reported by Fenaroli, okay? When they use a zebrafish model, they inject the zebrafish with a strain of Mycobacterium, which lead to the formation of granuloma after 4 days. Okay, and then they administer liposome. So after 4 hours, it's clear that the liposome accumulate at the granuloma site. Okay, so that is one of the way how we can deliver the nanoparticulate to the granuloma. Okay, so with the current technology, there are several research focus. Okay, so we want to improve the delivery of TB drugs. We want to reduce adverse drug reaction, and we want to improve patients' compliance. So this is among the main aims, okay, when we want to use advanced delivery system. So there are 3 possible approaches. This is the first one is nanoparticles. Okay, the nanoparticles is, as I mentioned earlier on, is a passive targeting approach. So we can use nanoparticles such as phytosan nanoparticles, okay, alginate nanoparticles, okay, to improve the delivery. So nanoparticles, following oral administration, it can be absorbed through the lymphatic system. So this will bypass the liver, and then it will directly enter the blood circulation. So this can be one of the way. Another way is by using GAPDH targeting. This is one of the study that we are currently conducting. whereby we look for targeting molecules that can target GAPDH. So GAPDH is an enzyme that is present on the Mycobacterium tuberculosis. So it's a receptor. Okay, so this receptor is important in the transportation of transferrin and lactoferrin needed by the Mycobacterium tuberculosis for its function. So when we can target this receptor, we can try to eliminate the Mycobacterium tuberculosis. Okay, and the third way is by nano FDC formulation. FDC refers to the fixed dose combination. So we can combine the nanoparticles formulation into a fixed dose combination. So this will help to improve patients' compliance. So I would like to share some studies that we have done. We are working on chitosan nanoparticles, okay, and also eudragid nanoparticles. So we have we employed the technology of the application of design of experiment to get the most suitable nanoparticle system with suitable physical characteristics. So we use a map, okay, that is generated by the design of experiment program, so that from there we can come out with the suitable particle size according to our application. The second type of nanoparticles and microparticles that we are currently working on is the graphene oxide. So we work on both graphene oxide and reduced graphene oxide. If you see at the bottom here, the left side is the graphene oxide and the right side is the reduced graphene oxide. So the graphene oxide has a more oxygen containing group NCOH, okay, at the perimeter here, but the reduced graphene oxide has less. So we compare the safety of these two carrier on the HS27 epithelial cells, and it is clear that GO is much safer than the reduced graphene oxide. So for human application, for future application, we are considering the application of GO for further development. And the one on the right here, the photo here is nanographene suspension. So it's brownish in color that we are working on to further develop in the future. Another study that we are also doing is on the identification of GAPDH targeting agent. So we found out that one of the useful molecule is folic acid that has a high affinity towards GAPDH receptor. So we do modeling works on how we can improve the delivery or the affinity of folic acid towards the GAPDH receptor. Okay, so finally, what is our aims? What do we wish to reach at the end of the day? So we would like to have patient compliance to the treatment that we want to give to the patient. We want to meet the target set by WHO to end TB by 2030 and beyond. Of course, we wouldn't want TB to come back. And of course, at the end, we want to also have the quality of life for the patient. We wish to have a good, we wish that the patient will have the life that they deserve, okay? And TB will not destroy anything that they have. So having a TB is not a death sentence, but in my opinion, as a researcher, we have the obligation to help or to contribute in what we can to meet the target set by WHO. So this is the aim. I know it's quite it's not easy to meet in the near future, but yeah, we are working towards that. So I would like to acknowledge the Ministry of Higher Education Malaysia for the grants that I obtained, and also my postgraduates and undergraduate students. Some of them are here that also joined this conference today. So with that, I would like to thank again for the organizing committee for the opportunity. Thank you very much.
Okay, thank you very much, Dr. Amira, for the speech. Thank you. Okay, Dr. Amira, we will meet again on question and answer section.
Okay.
Right now, we will move to our next speaker. Thank you so much, Dr. Amira. Our next speaker, Dr. Najiha, are you with us?
Yes, can you hear me? Can you hear me?
Yes, I can hear you. Dr. Najiha, can you hear me?
Oh, yeah, I can hear you. Sorry,
Okay,
I'm
good.
outside, I'm not in a room. So I
Oh,
hope you don't mind if there's any background sound.
Oh, okay. Okay, thank you so much for joining us today. We will give you time for 25 minutes and you can start your presentation. The time is yours.
Okay. I think better I share my slide first. Can you see my slide?
We couldn't see your slide yet.
Maybe the
Okay.
internet is slow.
We can see it now.
So is it in presentation mode?
Yes.
All right.
Not yet. Still freezing. Okay.
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 All right. Thank you so much. All right. Assalamualaikum and a very good morning to all participants and also to the organizer. I would like to convey my gratitude and appreciation and thank you to the organizer who has invited me to share all my research work with the audience. Okay, the title of my presentation today is about bioactive compounds from selected Malaysian medicinal plants. So I'm from Faculty of Pharmacy and I'm also the head of Center for Natural Products and Research and Drug Discovery in Musti Malaya. All right, so this is Malaysia. I believe everyone knows Malaysia. We are smaller, much smaller than Indonesia. So we have two areas, which is one here is Peninsular Malaysia and one is from the Kalimantan. So we are quite small in terms of size compared to Indonesia. So the size of our forest in total is about 20.1 million. So in Peninsular Malaysia, it's only about 6.2 million. I believe this is about 1 to 5% size of forest in Indonesia, right? So whatever it is, we are among the 12 mega biodiversity, in the world, rich with, plants, flora and fauna. Okay, so the flora of covering Malaysia, about 12,500 to 15,000 species. It comprising of the fern and about 1,159 and mosses about 560 species and more that we cannot estimate how many. And we do have many freshwater and marine algae which covering more about 1,400 species. So for this presentation, I will only share a few of my plants, a few of studied plants. We have a lot of plants that we study in but I would like only to share with you that two particular species that we have been studied with the active compounds we have managed to isolate it from these two plant species. One is from the Artocarpus species, where we have investigated about 13 plant species. So the one in yellow I'm showing here are the one that produce compounds that are active against a selected biological activity. So I believe Artocarpus species, they are family like jackfruit, cempedak, or nangka, right? I hope you know. Okay, next plant is the Catroxylum species. So from this Catroxylum species, we have isolated 3 compounds, but the only 2 compounds that showed very good activity. The one that been presented by Prof. Mutakridi, where the alpha and beta compounds can also be isolated from these plants, besides the mangosteen. All right, so these are the commonly, work done for natural product work, especially in isolation of compounds. So we extract the plants, we isolate the compounds and identify it using the spectroscopic methods. And then we do several testing, maybe at the preliminary screening. And then we do, which one is the best or potential, then we go for further isolations for active compounds. So for my work, for my group, we do in vitro study. So from the antioxidant assay, antimicrobial, and also in vitro assay against cancer cells. And we also did, previously, we also did in vivo study to test whether the compounds toxic or not to the cell or to the animal. So we do the acute toxic study, we do gastroprotective study, and also chemotherapeutic study. So for the chemotherapeutic study, we emphasize more on the breast cancer study. Okay, so these are the compounds that we have isolated from Artocarpus species. The one you see in yellow are the new compounds. These 2 are new compounds which I have isolated during my PhD. So they are new compounds been identified, isolated from this Artocarpus species. And the one that pyrosaccharatoglazantone A is the most active compounds, which I will show presented to you later. So these compounds are isomers, and they are only different in terms of the position of hydroxyl. So here you can see the position of OH at C14 and here at C13. So in terms of activity, they are also different, where the pyrosaccharatoglazantone, or I name it as PA, showed much better activity than this compound. So these are the compounds that have been isolated from Catroxylum aberrances, so alpha mangosteen and beta mangosteen, as well as fuscazantone. The alpha and beta mangosteen has been isolated in quite a large amount, so that's why we managed to do animal study for these 2. We managed to isolate about, I think, 15 grams from this plant, from about, I think, 2 kilos of plant samples. So I shall only going to represent to you these 4 bioactive compounds: the pyrosaccharatoglazantone A, or known as PA, adenine E, alpha mangosteen, and beta mangosteen. So the 2 compounds above here are the compounds being isolated from Artocarpus species, and these 2 later compounds are isolated from the Catroxylum aberrances. So this is the first compound, the PA, pyrosaccharatoglazantone. It is a yellow crystal. It's only dissolved in DMSO. The second compound that I showed you earlier, the isomer, it dissolved in acetone. So this is much more polar. And the molecular weight is quite big, it's about 450. And this is the first isolated from these 3 Artocarpus: Artocarpus obtusus, Artocarpus rigidus, and Artocarpus melanaxillus. So we have published this compound in Journal of Asian Natural Product Research. So the second compound is the adenine E. It's also yellow powder. It dissolved in acetone, and the molecular weight is 436. So it is isolated and been identified from Artocarpus elasticus. So this is flavonoids group. And now we go to the Catroxylum aberrances. So we have isolated alpha mangosteen and also beta mangosteen. I know that alpha mangosteen has been has a wide spectrum of biological activity, has been shown by Prof. Mutakridi, where it's a very good compound. And the problem with AM is about solubility. For this compound, it's hard to dissolve in some solvent. So we also published this some of the works in some of the good journals. So now I'm going to show you the work that we have done on anti-cancer. So let's see here. So we have screened the compounds, the PA, the pyrosaccharatoglazantone, and these 2 compounds against the least cancer cell lines. So the one that I highlighted in clear color here is the one that showed a very good activity against the cancer cell lines. So you can see PA showed a very good activity against leukemia cell. This HL60 and K56 are the leukemia cell. The HeLa is the cervical cancer, MCF7, MDAB, breast cancer cells, HT29 are colon cancer, COV and SCOV3 are the ovarian cancer. This is stage 2, the COV is stage 2, SCOV3 are the stage 3 with a metastasis ovarian cancer. And the rest 3 are the normal cell line. The T1074 are the normal cell line for ovary, MCF10 a normal cell line for breast, and PBMC is a peripheral blood monocytes. Okay. So this is also another asset value that we obtain after we screen against COV and SCOV3 for arginine, alpha mangosteen and beta mangosteen. So arginine show a very strong activity against these ovarian cancer cell lines, as well as alpha and beta. But we have not yet done any further study on alpha and beta mangosteen for ovarian cancer. So this is the work done by one of my students of arginine E and pyranosaccharidosis atoglazantone A against ovarian cancer, where you can see on the COV3 up here is a normal cell, a control, and below are the here are the treated. So you can see the number of viable cells are decreasing following the hour of the time exposure, and here is the PA also showing a very strong activity after the treatment against the COV3. And for this one, it showed the arginine E activity after the cell SCOV3 been exposed to arginine E, and it showed a lot of dead cells and less number of viable cells, as well as the treatment with PA against the SCOV3. So we again want to check whether the cell, the compound exert any apoptotic activity. So it shows here that the number of cells that been went through the apoptotic increasing according to time. So you can see the number of cell line that experience apoptosis are increasing when it reaches 72 hours. So it shows a sign of apoptosis. So you can see the sign of apoptosis is when the cell blabbing, okay, and you can see the chromatin condensation. So it shows that apoptosis is happening. So the orange color showing the apoptotic or necrosis, okay. So these apoptosis are important for cancer cell mechanism. That is a way of how the drug acts on cancer cells. So this is one of mechanism where we choose to study for our natural compounds in our lab. So we have published this work in PLAS ONE. All right, so this is another work again to confirm the pathways. Which pathway does this compound act on? So when we do this pathway caspase analysis, it shows that PA and arginine E has showing intrinsic activity against this cancer cell, against the COV, and also SCOV3. You can see the caspase 3 and caspase 9 is upregulated or elevated for arginine E as well as PA. However, for SCOV3, for PA, SCOV3 didn't show any significant upregulation of a caspase 8. So it shows that PA and arginine E only act through intrinsic pathway mechanism, and arginine E displayed both intrinsic and extrinsic pathway mechanism to the cells. So this is another work that we want to confirm, where we want to check which phase in the cycle does this compound act. So it shows that at G0 and G1, the compound exposed to arginine E be able to cause the cell arrest, and also at exposed to COV3 is also show cell arrest at G0 and G1, and for as well as PA. All right. So for the S phase, it shows that compound adenine E and compound PA also showed cell arrest at S phase. You can see after 27 hours, the number of cell as being slow. Okay, so now I'm showing you this is a 3D cell culture. Okay, for me, we actually plan to do the in vivo study, but then it's very expensive if you want to do nude mice, because to do ovarian, to induce ovarian cancer in animal, we need to use nude mice because we are using the cell from human. So it's very costly. That's why we think of to change to 3D cell culture. So we started to do the 3D cell culture where we grow the cell, which is mimic like in vivo environment using the 3D parametric. So we optimize the method. So when we look at when we culture the cell, it seems that it shows a very good growing of cell until day 7. When it started after day 9, it started to die the cell. So we start to culture until day 7. Then we do the treatment using all our compounds. So this is the activity that we get after we have exposed our drug to the cells, 3D cell culture. It shows that this is a comparison between 2D cell culture and 3D cell culture. So the result is very different. So it shows that in 3D cell culture, it shows more resistance towards the compound compared into 2D cell culture. So this is another work done by my student, another student who wanted to do in vivo study, who wanted to do the in vivo study for compound PA and also alpha mangosteen. For PA. So we tested also on PA and against the MCF7, it shows inhibition of cancer cells against the breast cancer. So it showed a very good activity of PA against the breast cancer. So we published this in several journals too. So alpha mangosteen also been tested on breast cancer. So it shows the inhibition of cells after being exposed to alpha mangosteen. So from there, we start to do the animal study where we induce the rat with LA7. So we induce it become, tumor to the gland, mammary gland. So we choose about 3 doses for the treatment. So this is chemopreventive. After we induced become a tumor, then we do the treatment. So we choose doses. That is the low doses and high doses of low dose of AM and also a high dose of AM. And also we compared with a control, positive control that is tamoxifen at 10 milligram. Okay, so it shows that a reduction of size of the tumor after we treated the cancer, the tumor with compound. So we can see a very different improvement of tumor where the size has decreased after treatment with 30 milligram and also 60 milligram. But 60 milligram show a very good activity as compared to tamoxifen. So these are the results that we obtained where you can see the reduction of tumor percentage. The number 3 here is AM with low dose and number 4 is high dose. So the number 5 here is with tamoxifen. So you can see there is not much different compared alpha mangosteen and tamoxifen when we use at 60 milligram dose. So we have another work done by one of my students also where I want to check whether this compound has the activity as a to treat peptic ulcer disease or any ulcer or gastric ulcer in the stomach. So we test with alpha and beta mangosteen. So we start to induce the animal, the rat with methanol. So how we induce it? We use ethanol, and then we do before we start the treatment. Before we induce the cell, induce the animal to become ulcer. So we do a chemopreventive. It's not a chemotherapeutic, but we do chemopreventive. So we will feed the animal first with the compounds, and then we feed the animal later with ethanol. Okay. So this showing you here is the ethanol without treatment, where you can see the blood here is so the ulcer, and the edema here in the histo examination. And this is after the treatment with proton pump inhibitor. So this is the result that we have obtained after treated with alpha mangosteen. So this is a control, and this is a treatment. This is still a control. So this is the one that treated with a positive control. So we wanted to be like get our compound to become less ulcer. So the next is here. So we treated with alpha mangosteen at 5 milligram and also 10 milligram and also 20 milligram. So you can see that at dose of 20 milligram, it shows a very good improvement for the ulcer. Okay, this is for beta mangosteen. So it's also show the same result where at higher dose, it show a very good activity. And also you can see at 10 milligram, it show a very good improvement. So this is the best dose for treatment because it show about 83.98% of improvement of the ulcer. So this is the results that we found from the activity. So it shows that improvement of the ulcer in the rat after treated with alpha mangosteen, and also there's no beta, it's not beta mangosteen here, but only alpha mangosteen. So another study, this is I think the last study that I would like to share with you, is antibacterial agent, where we screen all the compounds that we obtain the compound, the PA, against the pathogenic bacteria, and among all pH, a very strong activity against MRSA. This is hospital-acquired infection. Is obtained. The activity is about 20 millimeter inhibition zone, as compared to other compound is very, very strong, but it's not showing weak activity against some fungi. So this is another work done by one of my master student where he tested test the arginine E, one of the compound from Artocarpus, flavonoids, again the MRSA, the ATCC is MRSA, and also ATCBAA, this is Staphylococcus aureus, where it showed medium to strong activity for arginine compound, but 13.6 zone of inhibition, and MIC and MBC is quite low, where we also have published this work in +1. Okay, so in conclusion, we can say that Artocarpus and Carthusian species are very rich with phenolic compounds, and they are offering a lot of opportunity for development of pharmaceutical products, and also could be maybe API or active pharmaceutical ingredient, or could be new chemical entity. So it can give health, promote the development of drug to help the community in the world. So we have identified these 4 potential compounds: the pyranocycloactivation A, arginine E, and beta mangosteen, alpha and beta mangosteen, which are potentially to be developed as anti-cancer agent, gastroprotective agent, as well as antibacterial agent. So this is my last slide. So I would like to, convey my acknowledgement to the funders, University Malaya, the government, the Ministry of Higher Education, and the university who give the facility, and not to forget to the one who helped me a lot, the late Prof. Hamid Hadi, my supervisor previously, and my collaborators from UM, also from Jazan University, and not to forget my beloved student. Without them, I don't think we be able to get the good results. So these are all my postgraduate students. All right, yeah. Okay, do visit to our website. Yeah, I would like to, introduce about my faculty here too. If you want to know more about my faculty, Faculty of Pharmacy, please do visit our website at https pharmacy.um.edu.my. All right, I think that's all from me.
Okay, thank you so much, Dr. Najiha, for your speech, for your presentation. We will back to you on question and answer section. Right now, we will move to the next invited speaker. Thank you so much, Dr. Najiha. Okay, right now we invite Dr. Suman Sinha. Are you with us, Dr. Suman Sinha? please join the conference.
Hello, good morning.
Good morning. How are you?
Yeah, I'm good. Yeah.
Okay, Dr. Suman Sinha, please start your presentation. We will give you 25 minutes and the time is yours, please.
Okay, sure. Okay, so good morning, everyone. I'll be talking about application of molecular modeling and specifically molecular dynamics in understanding few protein ligand systems, which are important for two predominant diseases like cancer and malaria. Malaria affects a lot in this part of the world as well as in Africa. So I have done this work as a part of my postdoctoral fellowship with Tata Institute of Fundamental Research, Hyderabad, India. Although I'm currently affiliated to GLA University, India, but then I thought that it will be pertinent because to present this work here, because this conference is we have many like-minded people where we get to see many experimental studies. So my talk will be the application of molecular dynamics and specifically enhanced sampling as well as equilibrium dynamics towards understanding protein and protein small molecule diaspora. I've worked with Dr. Jagannath Mandal and Dr. Kalaneshwar Mandala as my two principal investigators, and both are very young and both are very enthusiastic. And with Jagannath, I have worked specifically on the galactin system, and with Kalaneshwar, I have worked more on the malaria system, AMA1-RON2 system. So my part one of the presentation will be a protein problem, where I'll be talking about malaria as well as the importance of these two protein partners called apical membrane antigen 1 and Roftenek protein 2. So this is the complex which forms, which actually is a deciding factor towards the erythrocyte cell invasion by the malarial parasite. So these two proteins they interact a lot, and then this erythrocyte membrane rupture happens, and then the malarial parasite comes into inside the erythrocyte membrane. So then we thought that is it possible that we intervene in this juncture, and then we try to understand how these two proteins interact first, and then comes, of course, how to interfere using molecules, therapeutic molecules. But the first part of my the understanding is how these two proteins are interacting. So these two proteins, AMA1 specifically, it has two domains, domain 1 and domain 2. In domain 1, there are few crystal structures in the protein data bank, but I have taken only 4 R19, and in all the crystal structures, we see that there is the domain 1 is stabilized by domain 2, which is also a kind of supportive finding from my study, which I'm going to present. And then there is a domain 2 loop which specifically guards the domain 1 part of the protein where the another protein partner called RON2 it binds. So there is a buried hydrophobic cleft which is being guarded by this domain 2 loop. So there are many hydrophobic residues which are this cleft is consists of many hydrophobic residues, and this cleft is actually guarded by this domain 2 loop. What happens is this domain 2 loop opens eventually, and then RON2 comes into picture. The other protein, which is actually a big protein, but the ectodomain of the RON2, which has been solved crystallographically, it comes and binds. So previously, a French group under the leadership of Professor Bollinger, they showed in one of their deciding papers that the domain 2 loop, which is actually somewhere here, is a deciding factor in the binding. As you can see, the green colored protein called RON2 is a 59 residue peptide, sorry, 39 residue peptide, which is the ectodomain part of the whole RON2, which is a 2000 amino acid protein actually. But this ectodomain, the ectodomain, the studies have been done with this ectodomain, which is a 39 residue peptide. This one, which binds with RON2. They showed that this group showed that the domain 2 loop is a deciding factor in this macromolecular recognition. As you can see, they conducted two sets of experiments. Experiment set 1 says that there is they studied the interaction when there is a domain 2 loop. And they also did experiments where they truncated this loop off, and they studied the recognition phenomena through isothermal titration calorimetry, where there is no loop and where there is loop. So they found that whenever there is the loop is absent, in that case the KD increases. In other words, the MA1 is able to hold the other partner RON2 tightly, and that's why RON2 escapes from the half-life of the complex is disturbed midway. So then we thought that is it possible for us to do and see things what is happening in the actual picture. Then then there was a problem in the crystal structure, because in all the crystal structures, because the loop moves a lot, so there is a lack of electron density in the crystal structures. In all the crystal structures of MA1, except one structure, which somehow could capture the looping position. However, so we have talked this, we have taken this crystal structure called 3ZWZ, and when there is a missing electron density in the place, so that becomes one of our scenarios. And the other scenario is then we another information here is this loop, the alpha helix of the loop, it completely coincides with the position of the RON2 alpha helix when present. So it is something amazing we found. However, so then I started to do MD simulations on the protein complex, which is MA1 RON2 when there is no loop. So 500 nanoseconds, 3 repeats. In all the repeats, I found that in the initial conformation, the alpha helical portion remains, and as time passes, the final conformation of the end-term helix was observed to be degrading. The end-term helix was getting degraded. That was one mind. So maybe domain 2 loop actually helps in stabilizing the alpha helix, if not stabilize the complete structure, the complete AMA1 protein. And then I've used MD Engine as a GROMAX version 2019, patched with Plumed 2, and I've used VMD and Primal and popular visualization softwares, and then I have used CharmGUI for system preparation. So then I mentioned earlier that there is only one crystal structure which has this PDB, the loop present in the PDB structure. So we took that structure of 627 and then we grafted this loop into, because the sequences are same. So we grafted this loop into 3ZWZ, and then we conducted simulations, many repeats of simulations we did. And then we found that the initial portion of which you can see in cyan, this loop actually is getting closed and interacting with the RON2 alpha helix, which is maroon colored here, and that we can actually see the loop is closing. That means the loop has some role in the arrest of the alpha helix peptide. And we see a lot of interactions here with GLN2024 from the alpha helix of the RON2. It is one of the most prominent interactions of serine 373. There's a hydrogen bond there. And then we saw the distance of the over simulations, and then we saw that these two residues comes a lot near to each other. And then we see the movie here. This is what actually happens. I don't know whether the movie will play or not, but yeah. So this is how this loop behaves, and this dangles a lot and explores conformations, and finally it comes on the RON2 helix and forms a stable interaction complex. So this is how it is actually happening. So then another thing also we found that this loop is not stable. No doubt is one of the reasons that this loop does not sit in a single place. It does not engage itself in a single conformation. It explores a plethora of conformations. But we wanted to know whether it is the limitations of the MD, because we cannot explore. We have limitations of time. Maybe this is a millisecond scale event, but in MD we could reach only till microsecond scale level. So then we wanted to know whether this loop is free energetically stable. Whether this loop is free energetically stable, and when it is coming to the closed conformation. So for that we did umbrella sampling simulations, and then we found that in fact when these I took this GLN2024 373 as a reaction coordinate in umbrella sampling, and I could find that when the distance comes down when they come to each other, towards each other, it's a free energetically stable conformation. This is a free energetically stable event. And what are the interactions that are forming? We saw that this isoleucine 2022, it actually gets trapped in, although for momentarily gets trapped in a hydrophobic subcleft by phenylalanine 379, 378, phenylalanine 385, methionine 374, and isoleucine 375, as well as from here we have tyrosine 142 and threonine 140. So it's a kind of hydrophobic, cryptic hydrophobic pocket. So is one of the reasons. And then we have lots of hydrogen bond interactions with many residues of the loop originating from the alpha helix of the RON2 peptide. And these are the two major interactions. Then the question comes, what are the important residues? What are the important residues from the RON2 helix, which are actually responsible for this macromolecular recognition from the helix, because from the RON2 helix, because in later stages, although I am not showing this data here, in later stages, we have plans and we are still working on developing inhibitors based on the RON2 structure. So 4 residues, GLN2024, GLN2025, and lysine 2027 and aspartic acid 2028. These 4 residues, they are most important residues according to the visual inspection and the hydrogen bond interactions which I have seen through my simulations. So then why not to see free energetically how they are responsible. So for that, we did what we did is we did again umbrella sampling for we mutated the 2024 to alanine. We one by one we mutated all these 4 residues to alanine, and then we recalculated the umbrella sampling potential. We found that although we could not capture for GLN2024A and 25A much, but we could actually find that this 2027A and 2028A are actually disturbing the system, where you can see in the x-axis the distance when this loop closes. So in a wild type system, we have the loop is closed in around 1.7, let us say, nanometer. And when it is mutated to alanine, the loop is getting closed at around 3.5 nanometer. In other words, the loop is not getting as closed as the wild type, which in other words means the loop is not properly getting closed. So that means these 4 residues, the interaction from the alpha helix of the RON2 peptide is actually responsible for this loop closing phenomenon. So most important residues are lysine 2027 and aspartic acid 2028. So then how to prove it? Can we design experiments based on simulation results? So accordingly, AMA1 was expressed. All the control peptides were synthesized. Control peptides like all these peptides which I have done simulation on, they were synthesized, and biophysical assays using surface plasmon resonance, which is one of the sorry, very much recognized assay technique were conducted. So some results, we can see that yes, KD, the native wild type RON2, the KD is, we could reproduce the KD as reported in the published papers, which is around 20 nanomolar. And then when we are mutating this 2024A, it becomes to 140 nanomolar, and then 2025A to 74 nanomolar. Surprisingly, as the simulations predicted, 2027A is 898 nanomolar, which is almost around 1 micromolar, and 2028A is 227 nanomolar. So in other words, these few residues, they're actually disturbing this phenomenon, and the peptides are getting aggressed fast. So what exactly happens when RON2 exists? So we have got a consolidated structure, so consolidated understanding that this loop must be doing something. So what this loop is doing actually? So for that, I did how this D2 loop helps retaining the arrested state of this macromolecular concept. this macromolecular complex. So for that, I did metadynamic simulations, well-tempered metadynamic simulations of six replicas. In these six replicas, I could see that this RON2, the peptide RON2, could actually come out of the complex, leaving the AMA1 behind, and this is what is actually happening as the loop actually keeps there, and after we apply bias, the peptide leaves the picture when as I can try to rotate it. I'm sorry for this, mostly due to internet connection. It's getting disturbed. So the loop actually tries to capture, it tries to arrest it, but it tries to hold the RON2 within itself, but this is a major function of the loop which we have understood. So we have submitted this work in a bioarchive. You can have a look here. You can have a look, and of course feel free to comment if you find it interesting, or you can also criticize the study. So can we use this knowledge to design better inhibitors? Yes, we are doing experiments to modify the structure of RON2 and doing mutations here and there, specifically doing mutations as in the alpha helix, and then we're trying to design new inhibitors. So which completes my part 1, and part 2 is how much time I have left. I have with me. Hello. Hello.
Okay.
How much time I have left?
I think about 5 minutes.
Oh, 5 minutes. Okay, fine. I'll just finish it. I'll fire away. Okay, so part 2 is a protein ligand problem in which I have worked on this protein called galactin-3. Galactin-3 is an interesting anti-cancer target, as well as it has been explored in Alzheimer's disease also. So in this work, we have galactin is there's a class of proteins which galactin 1, 3, 4, 10, 5, 8, 7, all these isoforms are there. So we majorly find that this active site is majorly conserved. So then we took galactin-3 as a prototypical ligand, prototypical system, and then we try to understand how these two ligands, LACNAC is a native ligand, is a cognate ligand, which is important for the functioning of the galactin-3, and there is a LACNAC derivative, which is an inhibitor of galactin-3, which has been developed and published somewhere in Journal of Medicine Chemistry. So we thought that how can we study these two protein ligand binding simulations using whole simulations, the binding simulations, and what are the insights we can get from these simulations? So it's a binding trajectory I want to show. So we actually took everything, water, ions, everything. They have been removed for clarity, and we tried to understand how these ligands actually interact in the protein pocket. It took us microseconds of unbiased simulations, and we could reproduce the crystallographic pose of the protein ligand system 3kgr, I mean to say, and this is the site which the ligand recognizes, which is actually the crystallographic site. So then, as you can see from the distance plots, both these ligands actually randomly explode conformations and finally could bind to the active site. And for both these ligands, we could actually reproduce the crystallographic pose. And as a result of that, you could also see that this Lachner derivative, as reported in the paper, is a more active compound because it has got a absolute free energy of binding, which is -5.7, compared to the cognate ligand, which is -3.8 kcal per mole. And as well as we could see that these the k off, which is the exit rate of the ligand, is very less. So there must be something which this ligand is doing, which has been not reported so far. So then we could see that there are interactions which are keeping both these ligands anchored to the active site, specifically from tryptophan 181 and tryptophan and arginine 162. And these are the two most important residues which create a lot of, as well as here arginine 144. Here arginine 144 doesn't play much role in the cognate ligand. However, the arginine 144 plays a lot of crucial roles in the design ligand. Then we could, as we understood the residues which are important for interactions, we mutated those residues to alanine, and we found that the protein just throws off the ligands upon simulation. These are equilibrium simulations where we could see that the ligands are getting released from the protein when these two residues are mutated, arginine 162 as well as tryptophan 181. And so these residues are important according to another important result from these simulations are we could catch the intermediate states upon one of the two major intermediate states from Markov state modeling, which we have got is two intermediate states of the coagulating ligand. This is the crystallographic pose which has been reported, and these intermediate state 1 and intermediate state 2 we could capture. And for the Lachner derivative, we could capture only one intermediate state. So is ligand residence time an important thing to ponder here? Of course, ligand residence time is an important thing to ponder here because we could, if we see the off rates of the Lachner derivative, is really slow. So the residence of the derivative in the protein pocket is more. So then we ran metadynamic simulations to understand the time differences, metadynamic time differences, of course. So yes, we could see that in keeping all parameters same, we could see that the ligand of the native ligand was leaving the pocket in 30 nanoseconds itself, but the design nanoseconds. But the design compound, it is taking more time to come out of these pockets. So minimum is 60 because we have a stochasticity in molecular dynamic simulations. So not all the simulations are same, but it is always at average what we see. The majority of the simulations, what says is a result for us. So, and then the other ones, they cross 100 nanoseconds and then they exit from the pocket. So yes, ligand residence time. Then what is this extra tetraphenyl ring, which is there in the structure?
Sorry, Professor, the time.
Yeah, I'm done. This is the last slide. Yeah. So this tryptophan 181, this extra tetraphenyl ring, it is interacting with the tryptophan 181 with the hydrophobic interactions and hydrogen and pi cation interactions with arginine 144 and as well as with arginine 162. It has pi cation interactions here. And so these are the major, we could publish this work to Journal of Biological Chemistry, and it was published last year. And you can have a look at the paper, and please revert back if you have some questions. So thank you so much. I am grateful to my supervisors, Dr. Kalaneshwar, Jagannath, as well as my late Professor Surijit Sengupta, as well as my postdocs in the lab, Dr. Jamshad and Dr. Jayakrishna Konairu, Dr. Chandramohan Pandey, and the PhD students, Anamika, Abhishek, and all of them. So thank you so much for the kind attention.
Okay, thank you so much, Dr. Sunan, for your presentation. I will invite again Dr. Amira and Dr. Najira to join the conference. We will move to question and answer section. Dr. Amira, Dr. Najira, could you please join us again here? And also Dr. Sunan?
Yes.
Yes, sure.
Yes, I'm in here now.
I will read the question. First question is for Dr. Amira. The question is, how about if the bacteria of tuberculosis have become resistant? Can we still formulate it to be the drug delivery system?
Okay,
Amira.
thank you. Thank you very much for the question. So as far as I know, and based on the available literature, if the mycobacterium has converted into MDR-TB, which is the multi-drug resistance, the current available drug is not possible to be used anymore. So we have to use a different drug, because when the MTP becomes resistant, they have a mechanism to overcome the drug activity. So it will not be active anymore. So even though we reformulate the drug into advanced delivery system, although it reached the bacterium, but it will not be able to kill it. So it's not possible. But one of my student in his research, he tried to see if by modifying the certain by conjugating the current available drug with other moiety, how can it help in the elimination of MDR-TB, or how can it prevent the resistance to develop? We do have research into that, but we have yet to come to a conclusion on that matter. But this study is more on simulation, molecular docking, molecular modeling, and not really on the physical, or the synthesis of the molecule yet. So we are still exploring the possibility.
Okay, thank you so much for the answer, Doctor. Okay, next question is for Dr. Sunan from Ms. Juliana Bacho. So the question is, from the many research results that have been, what bond have the most potential to be developed into the drug product or maintained so that the resulting product is better, especially in terms of delivery system, by looking at the result of the molecular dynamic data?
Delivery systems is a very good question. Delivery systems, I do not have much idea because I do not do pharmaceutics. I do not work on drug delivery systems. But of course, if you ask me what are the most interesting interactions which play dominant roles in protein ligand binding, there are two. Number one is hydrogen bond interactions, and number two, hydrophobic interactions. If you see the therapeutic profile, or thermodynamic profile of the drugs, it is always advisable to develop compounds or develop inhibitor molecules which are more towards the enthalpic side. That means more hydrogen bond interactions should be there rather than hydrophobic interactions. But generally for medicinal chemists like us, Because in my PhD, I did a lot of medicinal chemistry. Dr. Amira most probably remember me, and yeah. And but now I have become a hardcore molecular modeler now, because I always felt these things to be interesting, specifically molecular dynamics, where we can see the dynamic changes in the interactions. That is one of my major motivations to shift my research base. So hydrogen bonds, to answer your question specifically, hydrogen bonds are most important. So when you're designing a molecule, try to look for hydrogen bonds. How many hydrogen bonds the molecules are there? Try to do a small dynamic simulation and try to find out whether these hydrogen bonds are stable or not. So if they are stable, and then try to get a binding free energy. If it is good, is this binding free energy is originating from those hydrogen bonds? These are questions we should think, according to me. Yeah, I hope that answers your question.
Okay, thank you so much, Dr. Sunan. Okay, next for Dr. Najiha. Actually, the question is from me. I would like to ask about, because your research is about anti-cancer agent, since I also work in anti-cancer agent for my recent research, I would like to ask, the current challenge for development of anti-cancer research is the drug that cannot target it specifically for the cancer. I would like to ask, how can the natural product that you develop can like solve this problem or address this challenge? Could you hear me, Dr. Najiha?
Yes, sure. I can hear you clearly. Okay, so that is also our challenge here. So at the moment, what we working is only targeting for one mechanism first. So we work on one like apoptotic mechanism, and I also work on certain protein, like targeting to certain like KRAS protein. So well, that it shouldn't be at the beginning. So it should start with the virtual screening first. We have to go through this profiling, and then we can work in the wet lab. So that's how it should be. But working together with collaboration with a computational group is very important, where we can do the profiling, then later we can do the test in the lab. That's what we are doing now, because we have a lot of compounds, and we like just test against all the cancer cells. But then we have not yet target to anything. So we have now started to to target on certain proteins, so that it's going to be more selective to the certain cancer cell, and also the certain proteins. That's what we are doing now.
Okay, thank you so much, Dr. Najia, for the answers. Okay, right now we will move to the next section. We will give the certificate for all of you. Okay, the first for Dr. Sunan Sinha. This is the certificate as the invited speakers in our international conference.
Thank you so much.
Okay, the next one is Associate Professor Dr. Najiha.
Thank you so much to the organizer.
And last but not least, Dr. Amira Mohghazali. This is the certificate from us, from our university. Thank you so much for all of you.
Thank you very much.
Thank you so much for giving us your valuable time. Please stay with us. We will take a picture before we leave. Okay. 1, 2, 3. Okay. Thank you so much for all of you, Dr. Sunan, Dr. Amira, and Dr. Najiha, for your time. Thank you so much.
Thank you so much.
See you
Thank
again
you.
on other
Bye-bye.
opportunities. Thank you so much. Okay, ladies and gentlemen, here is the end of this section. Thank you so much for your attention, and I hope to see you again in another opportunity. I am Edwin, and thank you for having me as your moderator right now. Thank you.
Okay, thank you very much. A big round of applause for our moderator and the three of our invited speaker. The three of them is a which were with the topic is really informative and really a good topic for us. Okay, ladies and gentlemen, and the honorable distinguished guests, before we enter the parallel session, now we will enter the lunch break. And so for all the presenter in parallel session, we will meet again at 1 p.m. So please just prepare your PowerPoint for present. And in our parallel session, there will be divided in the 3 rooms in a, in a 4th room in a Zoom meeting conference with the room A, with led by apotheker Rina Andriani as from MSc, and then room B, moderated by Waode Ida Fitria as from MFOM, and the third one is a room C, led by Rismayanti Fauzia as from MSc, and the fourth room or the room D, moderated by, excuse me. Okay, Citra Dewi, S-Farm and Farm. Okay, again, for the presenter, please, we will meet again at 1 p.m. So please. Okay, 1 p.m. in waktu Indonesia Tengah. Okay, thank you very much. We will meet again. So here, we will be back again. Thank you. Okay, ladies and gentlemen, now we will continue our agenda for the parallel session. But before we start, I would like to ask for the presenter to change their name by name or the ID paper that we already accepted. And then for the moderator, please be seated on your screen. For the room A, led by Apotheker Rina Andriani SPAM MSc. Room B, moderated by Wode Ida Fitria SPAM MPharm. And then room C, moderated by Rismayanti Fauzia SPAM MSc. And then the room D, moderated by Chitra Aryani. Chitra Dewi, sorry, Chitra Dewi. As SPAM MPharm. So every presenter, please join your room based on your ID in room A, room B, room C, and room D. For this time being, all the moderator in each every room, the time is yours. Okay, ladies and gentlemen, thank you for all the speakers and moderator for the parallel session. Okay, sorry for the inconvenience. I will repeat now for all the ladies and gentlemen. Thank you very much for your attendance in our international conference in the second Mandala Waluya-International Conference on Pharmaceutical Science and Practice. Now we will announce the best presenter and the best poster for the second international conference. We will start for the announcement in the best presenter in the room A. Rani Rubianti with the ID OP6 from Poltekkes Kemenkes Tasikmalaya. Congratulations for Ms. Rani Rubianti. The second one, the best oral presenter in room B goes to Muhammad Zakwan with the ID OP17 from Institut Teknologi Bandung. The third one in room C, Ungsari Rizky Eka Purwanto with the ID OP19 from Stifar Semarang. And room D, Andri Kusmayadi with the ID OP28 from Universitas Perjuangan Tasikmalaya. Once more, congratulations for the names as the best oral presenter. Now I will announce for the best poster. It goes to Nur Herlina Nasir from Universitas Mandala Waluya. Congratulations. And for the placement in the third place is Himaniarwati. Okay, sorry, it's just joking. Okay. Thank you very much again. Thank you very much for the best presenter and best poster. Now we are coming to the end of our conference today. I would like to say thank you very much. Thank you for our sponsor, Bank Sultra, also Maxima Laboratorium Clinic. Thank you very much. All the best for you. And wassalamualaikum warahmatullahi wabarakatuh. See you in the third international conference, Universitas Mandala Waluya, on pharmaceutical science and practice for the next years. Thank you. Okay, for all the committee, we would like to ask you to invite you in the podium to take a picture together.